We analyze CD4 as well as CD8 cell population in activation markers, and what we assessed was HLA-DR as well as CD38, and we saw low levels of these markers, the dual positive for HLA-DR as well as CD38 in HIV2 as well as dual infected participants. But what was interesting is that the low levels of these markers, the dual positive for HLA-DR as well as CD38, looked at both plasma as well as T-cell immune responses. And what we have seen, looked at both plasma as well as T-cell immune responses. And what we have seen is elevated beta chemokine levels in HIV dual infected individuals. And some of these cytokines were actually negatively correlated with HIV viral loads in the HIV dual infected individuals. We have also looked at this, we have seen low central memory CD8 T cells with expanded effector memory subsets in HIV dual infected individuals. And we have also seen low expression of immune activation markers in HIV dual and HIV2 infected individuals. And lastly, we see that there's attenuated inflammatory responses in HIV dual infection as well as HIV type 2. And this actually coincided with what we saw in plasma, where we saw low pro-inflammatory cytokine levels. So to summarize up, we have demonstrated immunological differences among HIV2, HIV1 as well as HIV dual infected individuals that could underscore the potential mechanism that is potentially influencing disease, natural control and immune preservation. We have also shown that HIV type 2 and dual infection may offer immunological model for studying long-term immune non-progression. So I would like to thank my supervisors, Dr. Peter Kwashi, Professor George Che, and Professor Tumbi Ndungu, and our study participants, our teams at various hospitals and different labs where this work was done, WACCBIP, Research HIV Research Trust, Noguchi, as well as ARI for providing lab space for this work to be done. Thank you.
Thank you very much. We have time for questions. Yes, Dr. Agbawanyo.
Right, thank you very much. I was just wondering, you were kind of looking at is West Africa as well as Central Africa.
So
the
okay.
Samples.
West Africa. Not really West Africa, but Ghana.
Okay.
So our samples are coming from Ghana.
Right, okay. So I was wondering, would you say that you kind of noticed any spatial variations with respect to the cytokines from the different areas from which you collected the samples?
Okay, so our sample came from 6 hospitals in Ghana, and we have not looked at the geospatial differences among the samples. It's something we could look at and see if there is any. However, considering they are all West African population, it's likely that we will not see a lot of differences in terms of cytokine production. Yes.
Okay.
Yeah.
Okay.
Thank you very much for the wonderful presentation. I was just wondering if other populations you are looking at were on ART and the effect of ART on your parameters.
Yeah, they were all on ART, and they were for a period of about 1 year, 12 months. Yes.
Okay. Yeah, Prof. Monticello.
Quick one. The HIV2 dual and mono, I didn't see you profile CD4 T cells. I only saw CD8. Any reason?
Yes. So we did for both CD4 and CD8, but because of time, so we have result for CD4, but usually we do not see a lot of variations between HIV type 1 and HIV dual infections as well as HIV type 2. So we presented just data for CD4 for most of the assays. The effect of the CD4. So first of all, the CD4 were usually they're the target cells for HIV infection. So what we saw is that for the HIV dual infection, the CD4 numbers were a bit high compared to that of HIV type 1 because of the immune reconstitution after ART. But one thing that was interesting in the CD4 that we did not show here is that despite being infected, the CD4 seems to be producing more cytokines compared to the CD8. Yes. Yeah. Oh, okay, sure.
Okay, so I have a question.
Okay.
Yeah, so with the dual infections, does it matter which of the subtypes an individual gets first? And I was wondering whether you took that into consideration with your assays.
Yes, normally with the dual infection, when HIV type 2 comes in first, it kind of gives some levels of protection against getting HIV type 1. However, because we are working on a cohort of participants, and all of them already had been diagnosed as HIV positive, it's really difficult to tell which one came in first, because if you have to tell which one came in first, you have to kind of follow a HIV negative cohort until they get infected for the first time, is it HIV type 1 or type 2 that came in first. So with this kind of cohort, it's difficult to actually distinguish which of the HIV came in before the other. Yes.
I think we don't have any more questions. Thank you very much. So our next presenter is Mark Apieni. Is Mark around? Oh, online. So Mark will present online. All right, so you have 12 minutes, if you can hear us.
Yes, I can hear you.
Okay, please go ahead.
Just a moment. So good afternoon. My name is Mark Apieni, and I'll be presenting on how antiretroviral therapy attenuates key inflammatory pathways in people living with HIV in Ghana. So the HIV epidemic in West Africa is quite distinct. For we have HIV-1, we have HIV-1 and HIV-2 co-circulating, and more specifically, we have the circulating recombinant from AG being the most predominant HIV form in West Africa compared to what we find in other regions. We have the circulating recombinant from AG being the most predominant compared to what we find in other regions. In addition, we have a high burden of co-infection, including tuberculosis. Then again, we have a rising burden of non-communicable diseases within West Africa and Ghana. So putting this together, this complexity impacts treatment and the disease progression. HIV pathogenesis is multifactorial in that we have no one single immune change can fully explain the immune dysregulation that we find. Because, for example, we have cytokine switch. Some of them play a role in viral replication, and others are also involved in inhibiting the disease progress. The challenge here is that in West Africa, and particularly in Ghana, the immune pathogenesis is not well remain understudied. Knowing that there is a distinct HIV epidemic within Ghana and within West Africa, this underscores the need for localized immunological research. So the aim of this study was to investigate how ART modulates. The aim of this study.
Hello, Mark.
Was to
Is it
investigate
possible to increase your volume a little bit?
Okay. So the aim of this study was to investigate how ART modulates immune response among Ghanaian people living with HIV, and specifically to investigate how cytokine-driven mechanisms, cytokine-driven immune dysfunction, and to identify potential biomarkers for treatment response. So this is part of a bigger cohort study where we have already recruited HIV treatment naive, and we followed them up for 1 year, and we have HIV experienced participants who were on treatment for at least 6 months. We took blood samples, we measured viral load, then we measured plasma cytokines, 25 plasma cytokines, then we analyzed the data using R. So for the first part, we realized that HIV ART resulted in the decrease in most of the pro-inflammatory cytokines. So essentially what is happening is after the initiation of the antiretroviral therapy, there was there was a reduction in viral load, and this reduction in viral load is leading to decrease in inflammation and decrease in immune-mediated tissue damage, resulting in a decreased risk of non-HIV comorbidity. Again, we see a reduction in interleukin 15 and other T1 cytokines, and this is resulting in reduction in immune activation, and this prevents T-cell exhaustion. For the anti-inflammatory cytokines, with the exception of interleukin 13, we find a reduction in all the anti-inflammatory cytokines from our cohort. And what is happening is with reduction in inflammation, there's a decrease in anti-inflammatory compensatory mechanism coming in within our population. Then for the chemokines, we realize that for the chemotoxic cytokines, including MIT1 alpha and MIT1 alpha and CPR, there are reductions in their volume, and this is leading to a reduced immune cell capacity and reducing inflammation, and this is leading to immune stabilization. But for cytokine or for the chemokines that are involved in HIV entry blocking, especially irritation and rampage, we find elevated levels in these two cytokines. And what is happening is potential protection against viral re-entry, and this supports immune recovery among these participants. So furthermore, we wanted to find out if it's possible to predict the outcome of this, if it's possible to predict the outcomes of the disease based on these cytokines. So we used a proportional hazard regression to do this prediction, and we found reduced levels of interleukin 1 beta in 10 were associated with virologic control, while increased levels of granular macrophage colony stimulation factor was associated with viral non-suppressing. Then we wanted to find out how all these cytokines interact together. So we pulled out the data from string database and constructed a protein interaction, and selected the interaction with high confidence of 0.7. What we found is we found interleukin-1 receptor antagonist being central in all these interactions. Then again, we did a correlation to find out how all these cytokines were correlated with the viral load, and we found IP10 being positively correlated with viral load, whilst Ramptis negatively correlated with viral load. Furthermore, we wanted to see which pathways or which biological processes all these cytokines are involved in. So we retrieved the DNA of these cytokines we worked on, and we went ahead to do the gene ontology and KA pathway analysis. So what we found was, for the biological findings, we realized that for those cytokines, two cytokines that were elevated, Ramptis and eotaxin, they were mostly involved in eosinophil chemotaxis. They were mostly involved in chemotaxis, while those that were reduced were involved in leukocyte activation. Then we looked at the molecular function of these cytokines and chemokines, and for those two cytokines, chemokines that were elevated, they were mostly involved in chemotaxis, whilst the other cytokines that were reduced were involved in receptor binding, growth factor receptor binding. We went ahead to look at which of the pathways that these cytokines are impacting. Then we found out that those cytokines that were elevated, the ARC, were involved in cytokine signaling pathways and cytokine chemokine receptor interactions. While those that were reduced were mostly involved in interleukin-17 signaling pathway and JAK-STAT signaling pathway. So putting everything together here, we found that interleukin-1 beta, interleukin-10, and DSF were identified as potential biomarkers for disease progression. We found a situation where there is a suppression of interleukin-17 and JAK-STAT signaling alongside persistent chemotactic activity. So what is happening is the immune cells are actively being recruited, but they remain poorly activated or functionally impaired. So what we are saying is that the information from this study can be applied in immunotherapy, where these pathways can be targeted for adjunctive immunotherapy. And I would want to acknowledge my supervisors and all these other founding members. Thank you.
Thank you very much. Questions, please. No. It looks like we don't have any questions. Probably it was hard to hear the presentation, but Mark, are you still there?
Yes, I'm here.
All right, thank you very much for your presentation, and we'll move on to the next presenter.
Thank you.
So our next presenter is Comfort Kote. Yes. Her talk will be on differential IgG recognition of folded and heat denatured AMA1 proteins across 3 human Plasmodium species.
Good afternoon, everyone.
Good afternoon.
Please, can you hear me?
Yes.
Okay. So my name is Kote Comfort, and I'm presenting on the differential IgG recognition of folded and heat denatured apical membrane antigen 1 proteins across 3 human Plasmodium species in a malaria endemic population.
I think you have to. And you can look, Comfort, you can look through here too, yeah.
So malaria is a disease of global health concern, and in the year 2023.
Point it to the front, okay?
Okay.
Point it towards the direction.
But should I hold it this way?
Okay, so this is your
Okay. So, malaria is a disease of global health concern, and in the year 2023, the World Health Organization recorded 623 million cases and 594,000 deaths. Okay. Human malaria is caused by 5 Plasmodium species, and amongst this, Plasmodium falciparum is the most deadliest and the primary focus for most intervention tools and research efforts. Many tools have been instrumental in the fight against malaria, and this includes insecticide-treated nets, indoor residual spraying, and artemisinin-based combination therapies for treatment. Vaccines are imperative in the fight against malaria, and there are many leading stage-specific proteins that are being considered for vaccine design. One of these proteins is the apical membrane antigen 1 protein. It is expressed at the asexual blood stage of the Plasmodium parasite. And it plays essential role in merozoite invasion. And antibodies against PFAMA1 has been shown to inhibit red blood cell invasion.
So can you help him? Can you help
Forward. Yeah. Are you able to change? Yes. So they will change for you.
Okay. Please, the slide before this one. Okay, and it's also conserved across multiple Plasmodium species.
So say next.
Okay. So the apical membrane antigen 1 protein has an intricate fold, and there's a consensus that antibodies mostly target conformational epitopes, but then studies that support this claim are just a few. And contrary to that, other studies have shown recognition to both conformational and linear epitopes. It is therefore important intricate fold, and there's a consensus that antibodies mostly target, and we see a similarity of over 50% for all our proteins. So PFAMA1 is shown in red, PM is shown in yellow, and PO is shown in blue. Comparing PF and PM, we see a similarity of 52.1%. Comparing PF and PO, we see a similarity of 51.3%. And also comparing PM and PO, we see a similarity of 71%. This shows that these sequences are similarity, and we are seeing over 50% similarity. We also compare the structural similarity of all our proteins, and we see a TM score of over 0.5%. So a TM score is a template modeling score that is used to compare the similarities between two structures. So we see over 0.5%, which suggests significant similarities between the proteins that are being compared. Yes, we were also interested in quantifying antibodies against our proteins. So we first established a cohort with 116 samples from the Kintampo community, ages between 2 and 59, with 14 years being our median age. So we stratified our group into two: children below 14 years and adults over 14 years. And for our quantification, we treated one part of our proteins to heat denaturation at 80 degrees Celsius for 15 minutes. For the second part of the protein, we do not denature, and these two proteins are subjected to the same treatment, and then we quantified antibodies against them. Next slide. So this figure represents the distribution of antibody levels against our folded proteins and then our denatured protein. So we see high recognition to the folded protein as compared to the denatured protein across all our 3 Plasmodium species. Also, with our stratifications, we have the children and then the adult subgroups. So for the adult subgroup, we also see a higher recognition to the folded IgG as compared to the denatured protein. We also see that also for the children subgroup, there is a higher recognition to the folded protein as compared to the unfolded protein or the denatured protein. We are also interested in knowing the correlation between age and then the antibody levels. So we looked at the antibody levels against our folded protein with correlation with age. And here we see that as antibody levels increases, age also increases, or antibody levels increase with increase in age. So we see a moderate but positive correlation with IgG levels in all our 3 proteins. We also did the same for the denatured protein. We compared the antibody levels to the denatured protein against age of our study participants. And here we also see a positive but moderate correlation between antibody levels and age for both PFAMA1, for all our proteins, I mean PFAMA1, PMAMA1, and then POAMA1. Here we compared the difference in IgG levels between the folded proteins and the denatured proteins to age. This difference is what we are referring to as the antibodies targeting the conformational epitopes. So we compare that difference with the age, and here we do not see any correlation with age. Next slide. So that's what we call the conformational epitopes. So we compare the conformational epitopes to the linear epitopes, and we see that antibody levels against the linear epitopes are high compared to antibody levels against the conformational epitopes. And this runs through for all our proteins. Here we compared PF-AMA1, PM-AMA1, and PO-AMA1 IgG levels against the conformational epitopes. And we see a higher recognition to PF-AMA1 as compared to PM-AMA1 and PO-AMA1, suggesting that antibodies are more towards PF. And this is true because PF is the most prevalent Plasmodium species we have in our part of the world. Excellent. We are also interested to know if there's a correlation between the total IgG and we're also interested to know if there's a correlation between total IgG towards the folded protein and the conformationally dependent protein. So we have antibodies against the conformationally dependent epitopes on the y-axis, and on the x-axis we have the antibody levels against the folded protein. And here we see a positive correlation between antibody levels between the total IgG compared to the conformationally dependent epitope. So what we can take from this slide is that antibody levels against conformationally dependent epitopes increases as the total IgG towards the folded proteins increases. So in summary, we've shown that there's a moderate sequence correlation between the PF-AMA1, PO-AMA1, and then PM-AMA1 sequences and also the structures, suggesting potential cross-reactivity. Also, we've shown recognition of all our AMA1 proteins by naturally acquired IgG in a species prevalent manner. And also, we've shown that antibody levels are more towards the continuous or linear epitopes. And also, IgG towards the conformational epitopes did not increase with age. Thank you.
Thank you. Please, questions. Yes.
Very nice talk. Was there a possibility of you going back to the patients and seeing if there is a relation between the antibody levels and the plasmodium level or like the disease progression, so that, if you're thinking of this as a vaccine candidate?
With the disease progression?
Correct.
Okay, so these samples are some archived samples. So these samples are community samples. These people did not present with malaria at their hospital.
Okay.
Yes.
Okay. Thank you.
Okay. All right.
Just a quick thought. Why do you think more antibody concentrations to the linear conformation than the folded? What could be your basic explanation to that?
Please, can you come again?
Why do you think we observe more antibody responses to the linear conformation than the folded conformation?
Well, I can't really say what the reason is, but then this is what we are observing. Like I said in my introduction, other studies reported that antibody levels are towards the conformational epitopes. But then, contrary to that, other studies also showed similar recognition levels to conformational and linear epitopes. So, this is what we are observing.
Anyway, so...
Yes, Dr. Kwashi. Dr. Kwashi. Hello?
Okay.
My mic, okay.
Okay, yes, I forgot about online. Do you think it could be an issue of maybe sensitivity in detection for your conformational antibodies? So maybe there's an age-dependent detection, but you're not detecting that?
But from our study, we compared the levels, the IgG levels with age for the conformational epitopes, and we didn't see any correlation with age. Okay. So I don't think there was no correlation with age.
So I think we all agree that all the presenters.
Microphone for the online participants.
So I think we all agree that presenters did very well in this first section. Let us all give them a round of applause. I also think that lunch is ready and we will be told. Thank you.
Okay, thank you so much, Prof. Jira and Prof. Kwashi, for chairing this first session. We appreciate the timeliness and everything. And to our presenters who...
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of the department at the time, who was Prof. Saki, Prof. Samisaki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region, because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had, and their desire to learn, and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the thing. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, you know, Patrick, Lydia, Teresa, Osborne, Abida. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than $50 million for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honor for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the number one leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACCBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions. which have also been exported across the world. I think WACCBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the great continents.
WACCBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACCBIP has more than prepared me, and I wouldn't say this because I've just stayed in WACCBIP, but for the fact that WACCBIP has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WACCBIP was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented its Pandemic. WACCBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WACCBIP did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WACCBIP did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WACCBIP machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork and very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WACCBIP because the leadership of WACCBIP identifies young talent and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, schools engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WACCBIP has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WACCBIP has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups. Of course,
the idea of building private sector-based research to complement the academic research that is being built at centers like WACCBIP, I think is a natural evolution of the development of the center. WACCBIP's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work like what we do here?
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WACCBIP.
Happy 10 years, WACCBIP.
Happy 10th anniversary, WACCBIP.
Happy 10 years to WACCBIP.
Happy 10 years, WACCBIP.
Happy, glorious, and illustrious 10th anniversary, WACCBIP.
Happy 10th anniversary, WACCBIP.
Many congratulations to everybody involved in the WACCBIP story.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space. And also, that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department. And the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the thing. So I called Gordon and said, Gordon, I think you should host this.
So basically I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Theresa, Osborne, Abidad. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Ayite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordner Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than 50 million US dollars for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honour for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the #1 leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACCBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACCBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the great continents.
WACCBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACCBIP has more than prepared me, and I wouldn't say this because I've just stayed in WACCBIP, but for the fact that WACCBIP has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WACCBIP
was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WACCBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WACCBIP did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WACCBIP did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WACCBIP machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork under very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WACCBIP because the leadership of WACCBIP identifies young talents and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, schools engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WACCBIP has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WACCBIP has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups, of course.
The idea of building private sector-based research to complement the academic research that is being built at centers like WACCBIP, I think is a natural evolution of the development of the center. WACCBIP's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work that, like what we do here.
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WACCBIP.
Happy 10 years, WACCBIP.
Happy 10th anniversary, WACCBIP.
Happy 10 years to WACCBIP.
Happy 10 years, WACCBIP.
Happy, glorious and illustrious 10th anniversary, WACCBIP.
Happy 10th anniversary, WACCBIP.
Many congratulations to everybody involved in the WACCBIP story.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region, because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the wing. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, you know, Patrick, Lydia, Teresa, Osborne, Abida. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than 50 million US dollars for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honour for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the #1 leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACCBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACCBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the great continents.
WACCBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACCBIP has more than prepared me, and I wouldn't say this because I've just stayed in WACCBIP, but for the fact that WACCBIP has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WACCBIP was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WACCBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WACCBIP did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WACCBIP did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WACCBIP machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork and very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WACCBIP because the leadership of WACCBIP identifies young talents and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, cruise engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WACCBIP has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WACCBIP has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups. Of course,
the idea of building private sector-based research to complement the academic research that is being built at centers like WACCBIP, I think is a natural evolution of the development of the center. WACCBIP's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work like what we do here?
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WACCBIP.
Happy 10 years, WACCBIP.
Happy 10th anniversary, WACCBIP.
Happy 10 years to WACCBIP.
Happy 10 years, WACCBIP.
happy, glorious, and illustrious 10th anniversary, WACCBIP.
Happy 10th anniversary, WACCBIP.
Many congratulations to everybody involved in the WACCBIP story.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities. That was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the wing. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, you know, Patrick, Lydia, Theresa, Osborne, Abida. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than 50 million US dollars for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honour for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana. I wanted to work in an area that had a huge impact. I mean, there are no practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the #1 leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACCBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACCBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the great continents.
WACCBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACCBIP has more than prepared me, and I wouldn't say this because I've just stayed in WACCBIP, but for the fact that WACCBIP has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WACCBIP was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WACCBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WACCBIP did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WACCBIP did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WACCBIP machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork under very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WACCBIP because the leadership of WACCBIP identifies young talents and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
we have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, schools engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WACCBIP has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WACCBIP has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups. Of course,
the idea of building private sector-based research to complement the academic research that is being built at centers like WACCBIP, I think is a natural evolution of the development of the center. WACCBIP's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work like what we do here?
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WACCBIP.
Okay, as we're getting ready for the next session, if you know somebody that's outside, just let them know that we're getting ready for the next session, which will start in, I think, about 5 minutes time. So I hope everybody enjoyed their lunch. Not too much though.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region, because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the ring. So I called Gordon and said, Gordon, I think you should host this.
So basically I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Theresa, Osborne, Abida. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordner Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than $50 million for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honour for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the #1 leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACCBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACCBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the continents.
WACCBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACCBIP has more than prepared me, and
Good afternoon, everyone. I hope everyone enjoyed their lunch. And again, like I said earlier, if you know somebody who's not in the room, please text, message, let them know that the session is starting. And we're going to have this next session of the day, which is host-pathogen interactions. And I'll just introduce the chairs for the session. And that will be Dr. Frederica Partey, who is at the Noguchi Memorial Institute for Medical Research, and our very own Professor Osborne Quay, former head of Department of Biochemistry, Cell and Molecular Biology, and also one of founding members of WACCBIP.
Hello. Welcome to the first afternoon section after lunch. And as mentioned, there is the host pathogen interaction section and the second section for day 1. So I'll hand over to Frederica for the introduction of the first speaker.
Hello. Hello. Okay, good. Yes, so we have 2 talks this afternoon, and we'll start with one from Elvis, who is an alumnus of WACCBIP, and he would be talking to us about biallelic Marvel D2 variant identified with exome sequencing in a consanguineous multiplex Ghanaian family, segregating non-syndromic hearing loss. Elvis, take us.
Thank you very much. Good afternoon, and thanks to the organizers for giving me this opportunity. So as mentioned, I'm Elvis Chumesi Abuaje, a product from WACCBIP, currently at the University of Cape Town doing a postdoctoral fellowship. So I will talk to you this afternoon on a variant that we identified in a Ghanaian family that is segregating post-lingual non-syndromic hearing impairment. So when we looked at the burden of the condition, hearing loss or deafness, as I'll be referring to throughout my talk, the condition is a heterogeneous and very alarming. Currently, we are looking at close to 500 million people affected worldwide. And as you can see from the chart, the trend keeps increasing, and estimates shows that if nothing is done, we are looking at hitting as high as 900 million people to be impacted by this condition. The implications of this condition or any auditory disorder defect spans through communication challenges. It can impact speech. It can also language development, education, as well as employment. When we also look at emerging studies, it has been demonstrated that early detection, accurate diagnosis, and timely intervention in individuals that are affected actually improved the well-being of these affected individuals, improving their quality of life. So if you're able to diagnose it early and timely intervention provided, we are likely to make some headways. But unfortunately, on our part of the world, particularly when it comes to non-syndromic hearing impairment or hearing loss, the diagnoses are usually missed, they are delayed, and in most cases diagnosed late, at which point any intervention that is provided doesn't yield any impact in the quality of the affected individuals. So the team here, the group headed by Prof. Gordon Awandari, here actually reported this particular systematic genetic investigations that we conducted in families that are affected with this condition across the country. And as you can see, it shows very high genetic diversity when you look at the genes that have been implicated. If we are looking about 51 families, and we identified 4 candidate genes that have not been implicated in the condition, and 2 novel variants that were identified in known hearing impairment genes. So this afternoon I will take you through what I've highlighted here, a gene that's a variant in MAVEL containing domain 2 that we associated with the condition in a consanguineous family. So as you can see, when you take a look at the family structure, the whole exome sequencing analysis identified 3 affected siblings that are homozygous for this mutation in the MAVEL containing domain 2 gene. When we look at all other individuals that are not affected, they are either carrier, like the IV2, or homozygote of the wild type. So when we did the genetic and phenotypic correlation, looking at the audiograms, whether these individuals actually can hear different sounds, sound at different thresholds, we can see from these audiograms that the unaffected individuals have normal hearing. The threshold of hearing graded from -10 to 110 in decibels. So if an individual can perceive sound within -10 to 25, then that individual has a normal hearing. So as we can see from our audiograms, those 2 sampled individuals from the family, that one is a carrier and one has the wild type of that particular allele, have normal hearing, compared to the 2 affected individuals that showed profound impairment. So the whole exome sequencing results was actually validated using Sanger sequencing, as you can see in the chromatograms, where we can clearly show the duplication of this variant at that position for individuals that are homozygote for this variant and carrier, as well as the wild type individual. So to further investigate or interrogate this variant, we perform some in silico analysis, and as you can see from the 3D structure of the protein, the wild type in gray, the mutant in red, and the superimposition to actually see what is happening. So the variant causes a frame shift that results in the truncation of the mutant protein. So this can clearly be seen from the structural analysis. The mutation actually truncates this cytosolic domain, C-terminal domain of the marbled protein. And also, when we performed conservation analysis using 20 different species amino acid sequence for that conserved domain, clearly shows that the valine at that position is conserved in all the species that were investigated. For our individuals or for the affected individuals, we can see that the valine has been substituted for serine, and this actually leads to a truncation after 4 amino acid sequence. Database search also shows that the variant is indeed rare, and even in controls that we investigated in our population, as well as the cohort of both isolated and multiplex cases that were investigated as part of the cohort, the variant was absent in these individuals. ACMG classification also confirmed that classified the variant as pathogenic. So I will take you through some cellular work that we actually followed up to do to further confirm the pathogenicity of this identified variant. So we used cellular construct to actually investigate the predicted truncated mutant protein to infer molecular and biological significance of this particular variant. So we actually had 2 constructs that were generously gifted by Prof. Crook at the University of Berlin, which had a full-length human marbled sequence, perfect for our cellular work. So we moved on to create the variant of interest using site-directed mutagenesis, after which the variant was, the sequence was validated using Sanger, and we now moved on with the plasmid DNA for our cell work. So first, we wanted to confirm whether indeed the truncation or the nonsense-mediated mRNA decay that we are predicting as the likely cause for the mutant protein association to our phenotype. We performed Western blot with the first plasmid that has a flag tag. So as we can see, the wild type showed a prominent band compared to the mutant. That gives about 37 kilodaltons compared to the wild type that is 64 kilodaltons. Clearly, at the position where the truncation occurred, we are looking at losing approximately or close to 100 amino acids for that domain that has been lost. So the Western blot actually compared to the internal beta-actin control that was used showed that indeed the variant or the mutation results in the mutant protein truncation. So before that, to further investigate this MAVELD protein, studies has shown that the MAVELD containing domains interact with scaffolding proteins, occludin 1 as well as other membrane associating proteins to form complexes. And this localize at tricellular tight junctions to actually regulate the integrity of cell membranes or cells, to make sure that the cells are intact, they don't lose ions, they don't lose some molecules. And this has been key. This is keenly played when it comes to the hearing. So we moved forward to investigate whether the loss of this C-terminal domain will actually impact the binding activity of our mutant protein compared to the wild type. So we performed a pull-down assay to investigate this. However, the pull-down assay showed that not much was actually happened, not much difference between the wild type and the mutant. The interaction that we anticipated, that due to the loss of this C-terminal domain, may prevent or may cause no interaction with the membrane-associated protein, it was same for the wild type and the mutant. This clearly suggests that there's likely some redundant proteins or some compensatory proteins that has generated further questions to be answered. We moved on to actually use the other construct that has a fluorescent tag to still quantify or check the expression levels between the wild type and the mutant. And 24 hours post-transfection with HEX cells, we can clearly see the wild type up the expression intensity compared to the mutant. 48 hours, we can clearly see a clear difference between the expression that we obtain from the wild type compared to the mutant. We moved on again to look at cell morphology and to see the localization. And as we can see, when we compare the wild type to the mutant, whilst in the wild type, the localization actually, I must say, appeared as cytoplasmic inclusions. And whilst we can clearly see dense expression or dense fluorescence, when it comes to the wild type, the mutant looks like sequestered and also patchy when we compare these two. The same was observed in another one. Then to also look at this, our protein of the MAVELD interaction with the cell membrane and its integrity, we try to perform immunocytochemistry to investigate the actin filament complex that the MAVELD is known to interact with other scaffolding proteins. So we performed immunocytochemistry studies, and the results, as we can see here.
Alice, you have 3 minutes more.
Okay, I'm almost done. Thank you. So the results from the immunocytochemistry. So the immunocytochemistry, we're targeting, we use phylodin. Phylodin is to stain the actin filament, the cell cytoskeleton. Then we have our fluorescent protein. Then we also have the zonular occludin, that is the protein that our protein is known to interact with. So when we look at the panels, with the phylodin, we can see, comparing the wild type to the mutant, we can see a slight difference where when we look critically at the wild type cells, the rounding is consistent, which does not suggest any impaired membrane integrity. We looked at the ZO1, which it's known to interact with our target protein, which is also, we can see, highly concentrated or highly dense when you compare that one to the mutant. What is key here is that the protein is known to interact with this ZO1 to localize at tight junctions, so particularly tricellular tight junctions. So when we compare the two, we can clearly see that there are much spaces or a bit wider gap when you compare the positive patches of the ZO1 interaction with our target protein in the wild type compared to the mutant. So another view also shows the density in the expressed tagged wild type protein compared to that of the mutant. And these cortical acts and stress fibers are consistent with the expression of MAVEL2 in vitro studies. So in summary, from this cell assay or from this cell experiment, we've been able to demonstrate that indeed the identified MAVEL biallelic MAVEL-D2 variant, this frame shift variant, it's likely the causal variant that has been linked, that was linked, associated with the development of the condition in this consanguineous family. In addition, we can also say that it's likely that the nonsense-mediated mRNA decay mechanism of cells that it's used to protect or degrade aberrant protein synthesis from the cells actually is the likely mechanism that takes care of the mutant protein. And even if the mutant protein is expressed, it's likely to be expressed at a lower level. And even coupled to that, there's also the likely truncation of the C-terminal domain. We also believe that this is likely to impair the localization of this MAVEL protein at tricellular tight junctions, to which can compromise the membrane integrity, particularly when we talk about the inner ear, where it is critical for the homeostasis of key ions in the inner ear space. So the emerging questions from this particular assay is to further interrogate the redundancy and the compensatory mechanism and look because of the sequestration appear like Golgi sequestration structures that were obtained for the mutant, we are looking forward to actually label the Golgi's, label our experiment or stain our experiment with Golgi specific markers to actually see what is going on there. And also there is also a high likelihood and chance of actually using organoid models, getting disease-specific models to further interrogate this particular variant. I would say I'm grateful and honored to be mentored and supported by this great man. I will begin with Prof. Gordon Awandari. Then Prof. Susan, she is the person that donated the construct for us to be able to do this work. And I thank everybody. Thanks for listening. Thank you very much.
Very nice talk. In your slide number 6, if you're looking at the immunoprecipitations, there's a 180 kDa band in the mutant transfected plasmid cells.
Come again, please.
In your slide number 6, would it be possible to go back to that slide?
Yes, the immunoprecipitation.
Yeah, so you said that there was like no difference in the ZO1 IP by wild type as well as mutant, but that in the mutant transfected cell, I said, you have a 180 kDa band in the flag with the flag antibody. That's lighting up with the flag antibody.
So what? So what I?
Yeah, so in like the flag western, yeah, in the lower panel, yeah, that 180 kDa band.
So ideally, this is a pull down assay. So it's you are targeting specific interaction or specific binding. Mm-hmm. So the flag is like our positive control, and the non-specific IgG, it's the negative control. So the target is for this zoo interaction. So if the wild type is having the interaction, we don't expect the mutant to have the interaction because I've already indicated that it's based on this particular literature that this particular domain has been well characterized and it's known to interact with the ZO1. So it's that interaction that the pull-down assay was specifically designed to actually investigate. So now, because this is an ectopic expression in cells, we are looking at when it comes to the human ear, or when you get models, are there any other compensatory or redundant proteins that are also interacting, that might be interacting with this to give us this particular result?
Okay, we can talk later, yeah.
Okay, do we have any more questions? Okay, I see Craig.
Do your patients have any other phenotype other than hearing loss?
So thank you very much for the question. So these are 3 siblings. So these are 3 siblings that they developed the condition post-language acquisition. So they were actually speaking before they now went deaf. So the first, the proband actually developed the condition at age 12, and the 2 other siblings, the sister and the brother, at age 8. So aside the hearing, they do not have any other syndrome attached to, associated with the phenotype. So it is interesting because there's even some slight inter-family variability when it comes to the phenotype expression that we are also considering looking at.
Okay. So I have a quick question for you. So in your introduction, you mentioned that you had identified 6 genes associated with non-syndromic hearing loss, and 2 were novel. So is the marveled part of the novel, or part of the 4 already known ones?
Yeah.
And then a quick question is, I know you are trying to understand the mechanism of action, but what are the implications of what you are doing to enhance diagnosis? Because that's like one of the major problems we have.
Thank you very much. So I'll touch on the diagnostics first. So from the work that was actually done as on this particular investigation, we have actually, my colleague, Dr. Maoli Adade, together with a team, led, and we have developed a molecular diagnostic tool, which is very specific and sensitive, but it was first developed to target the most predominant cause of the condition among Ghanaians. So there is a particular gene that when we take a look at the Ghanaian population, whether it's isolated or whether it's a multiplex family. When I say multiplex and isolated, what I mean is with a multiplex, it means you have more than 2 individuals in the same family affected with the condition. When I say isolated, and I'm referring to families that had only one person that is affected. So it can be a spontaneous mutation that developed in that individual, the person didn't inherit it. So this particular molecular diagnostic tool is a simple PCR and RFLP that we've actually designed, available. Prof actually led the team to develop a policy document. The policy has been published, and we have a policy document that has been submitted to the Health Ministry, to Ghana Health Service to actually we are waiting to follow up for that is in line with the diagnosis that you mentioned. But for this particular marveled D2 variant, as I said, it's a novel variant in a known non-syndromic hearing impairment gene. So worldwide, we have about 125 different genes. So the condition is highly heterogeneous. 125 different genes have been implicated as causing deafness. But when we zoom in into regions and populations, although one gene, the gene that I mentioned, gap junction beta 2, so it's a gene.
Okay, thank you. I think I will see you at the top just so we can save some time.
Okay.
Thank you.
Thank you.
Thank you very much, Elvis. I guess we'll go ahead with our second talk, and this will be by Helene Kamasa. She'll be talking on microbiome signatures in human papillomavirus positive women, reveal links between vaginal dysbiosis and cervical dysplasia. Helene, over to you.
Thank you, Prof. Good afternoon, everyone. My name is Helene Kamasa, and I'm very pleased to share with you my latest research output titled Microbiome Signature in HPV-positive Women Reveal Links Between Vaginal Dysbiosis and Cervical Dysplasia. I hope you can hear me clearly. Okay, thank you. So, any help? Thank you. Okay. Thank you, Alfred. So cervical cancer is the 4th most common malignancy in women worldwide, with an estimated over 300,000 new cases and over 300,000 deaths per year. This, unfortunately, 95% of these cancer fatalities occurs in developing countries, with the greatest incidence rate recorded in sub-Saharan Africa. Hey. Oh, sorry. Okay, so yes, I can continue. This cancer is mainly caused by persistent infection with HPV infection. Actually, there are over 200 types of HPV, and 40 of them has been identified and classified as low risk, high risk, and moderate risk HPV based on their clinical oncogenicity. The normal vaginal cervix is made up with stratified squamous cells that range from the basal to the apical superficial layer and normally resistant to any viral infection. So there are microabrasions that occurs during sexual activity that enable the viral entry and the infection of this basal keratinocyte with the HPV. So the virus multiply actively within this basal keratinocyte and express the oncogenes such as E6 and E7, and upon different differentiation, the final differentiation, the viral particles are released, and this special mechanism help HPV virus to evade the host immune response and continue to be a non-explored infection. So what is known is that the majority of women can be infected with at least one HPV genotype in their lifetime, but fortunately enough, 90% of them can clear this infection, while 10%, the infection can persist in 10% and cause cervical dysplasia and even invasive cervical cancer. So the remaining question is that it is not known why some high-risk HPV resolve clinically, while others persist and can cause dysplasia and invasive cervical cancer. Emerging evidence has shown that vaginal microbiome can be associated with cervical cancer. So here we hypothesize that alteration in microbial diversity and composition may contribute to cervical disease. So to achieve or to test this hypothesis, we aim to characterize the vaginal microbiota composition associated with high-risk HPV infection and cervical cytological abnormalities in women, among women in Togo. So specifically, we aim to investigate the high-risk HPV genotype associated with cervical dysplasia, and secondly, to identify the vaginal microbial diversity and community composition among these women. So after the ethical approval was granted here from Ethic Board of University of Ghana and Togo Health Ministry, we use an advertising-based approach to convey participants through social media and radio to attend a cervical screening program. So once there, a focus group discussion was performed and the study aim was explained to the participants. So those who were willing to participate signed an informed consent. So overall, we enrolled 474 women across over 50 communities in Lomé, the main town of Togo. Each of them provided a cervicovaginal specimen that we use to perform a pap smear evaluation, high-risk HPV genotyping, as well as sequencing, and the data was analyzed using R. Overall, we saw 10% of cervical dysplasia in these apparently healthy women, and this is alarming. So we also saw an overall prevalence of over 68% of HPV, while we have HPV 16, HPV 35, and HPV 51 being the most prevalent among these high-risk HPV. When we stratify the data based on the cervical dysplasia status, we still saw a high prevalence of high-risk HPV among both cervical dysplasia positive and negative women. And conversely, we didn't see HPV 39, HPV 51, 56, and 59 in the cervical dysplasia positive women. So This actually highlights the differential oncogenic potential of some high-risk HPV and suggests that even though HPV is a key driver for cervical disease progression, this condition is not sufficient enough for cervical disease progression, suggesting again that other factors might come into play. So to further explore these other factors that can explain this condition, we performed a 16S ribosomal RNA sequencing, and the data was analyzed using R, where we computed alpha and beta diversity as well as tarsal composition. Alpha diversity is one of the metrics that are used to evaluate microbiome. So we evaluated this alpha diversity across HPV positive and negative group. And as you can see here, we didn't see any statistical difference. And the same trend was also observed when we compared the alpha diversity matrix across HPV positive, cervical dysplasia positive and cervical dysplasia negative women. And this data actually aligned with other findings that saw a stable alpha diversity in vaginal microbiome. We also went ahead and evaluated the beta diversity using a Bray-Curtis dissimilarity index. This index actually ranged from 0 to 1, and the higher this Bray-Curtis dissimilarity index, the higher the dysbiosis condition of the microbiome. As we can see here, most of the communities we evaluated are almost 1. So after comparing the Bray-Curtis dissimilarity index in HPV negative group compared to HPV positive group, we saw a statistical difference. And this difference is even more pronounced when comparing cervical dysplasia negative and cervical dysplasia positive women. So it is known that dysbiosis can induce chronic inflammation that can accelerate neoplastic change even independently of HPV infection. Furthermore, we try to visualize the bacterial communities using a principal component analysis. So we didn't see, even though the Bray-Curtis dissimilarity index show a statistical difference, we didn't see any difference. We didn't see any difference when comparing the vaginal bacterial community across HPV positive and HPV negative individuals. But we saw a soft difference here in cervical dysplasia positive women compared to cervical dysplasia negative women. So to further evaluate the bacterial tassa that drive this difference, we computed the relative abundance at both phylum and genus level. At the phylum level, we saw that Firmicut is actually
Helen, you have 2 minutes more.
Is actually distributed across all these HPV positive and HPV negative individuals. So we saw a potential decrease in HPV positive women for Firmicut, as well as a prominence of Actinobacteria and Fusobacteria in HPV positive women. But at the genus level, we saw a reduction of Lactobacillus in HPV positive women and an increase of Gardnerella prevotella, those ones who are actually anaerobic bacteria that are known to increase or to secrete other microbial enzymes that can contribute to the cervical dysplasia, cervical epithelium disruption. So the same thing was, the same thing was also performed for cervical dysplasia positive and cervical dysplasia group, and it was the same trend. So as a summary, this study has shown that specific microbial signatures such as lactobacillus depletion and enrichment of anaerobe may influence host susceptibility to HPV and the development of cervical dysplasia. I know that everyone here has a female relative, or even if there are clinicians here. So the take-home message is that HPV screening alone is not sufficient for cervical disease prevention. So we should also consider the macrovaginal microbiome profiling as a potential target for this condition prevention. So I would like to acknowledge my supervisors, WACCBIP and GSK for the finding. Thank you.
Any question for Helene, please? I will allow two, because we ran out of time.
Have you looked at women that sell roasted plantain and cervical cancers? Because of the heat, that will destroy the microbiota of the vagina.
We didn't get the question.
Sorry, the question again.
I'm asking whether is there any study of women who sell roasted plantain and cervical cancer because of the heat that gets into the vagina and might destroy the microbiota of the vagina?
Unfortunately, I cannot hear the question clearly. Prof, is it possible to relay the question?
If there is any study showing a link between women who roast plantain or who are very close to high intense heat and HPV or cervical cancer.
Okay, so actually, from the literature, I didn't see any study that explore this association. Thank you.
The hair. Okay, go ahead.
Thank you. Thank you very much for that excellent talk. As you mentioned, bacteria microbiome might also associate with HIV to produce cervical cancer. But I would like to know if you explore also immune inflammation in general, because I think that's also like a causality to cervical cancer, and by measuring like immune inflammation marker in those biopsies or sample that you collected. Thank you.
Okay, thank you very much for that question. Effectively, we did that. This is another component of this project that is ongoing, where we explore the cytokine, the pro-inflammatory and anti-inflammatory cytokine, local vaginal cytokine in this woman. So we have also this data, but because of time, we cannot present all the data here. Thank you.
Microbiome. How does it align with the different microbiome?
Okay, thank you. So with the women that have... Oh, okay, thank you. Okay, thank you, Doc. So with the women that have HPV infection and also bacteria anaerobe that are known to increase inflammation, we saw a high level of some local cytokines such as IL-6, IL-12, TNF-alpha, and so on. Yeah. Thank you.
Yes, thank you. Very nice presentation. So I'm wondering, with you've shown that lactobacillus levels are low, and we also know that so that would imply that the pH of the vagina would also be perhaps increased. So I'm wondering whether that is something you are also looking at. And also, I'm also wondering, in your work, are you also exploring perhaps if some of these women are also using some agents into that environment for various things that could alter the pH? Thank you.
Okay, thank you very much for that question as well. So we also collected other socio-demographic factors that come into play for the risk factors of cervical cancer. So we check whether these women perform things like vaginal douching and all those ones. So, but in this cohort, we didn't see any woman that is performing vaginal douching. Yeah, so the pH is also another factor, but unfortunately, we didn't check the pH. So maybe we can consider this one for further studies. Thank you.
Thank you very much. I guess that is all what time will permit us. You can engage Helen after this section.
Thank you.
Before we depart the podium, understand it is lost and found. So if you left your diary at the lunch area, okay, all right. Okay, the faculty area, okay. So then please see somebody for identification and collection. Thank you.
Please help me to appreciate our chairs. So we have a slight change to the program. We're supposed to have presentations from some of our gold sponsors, and they've indicated that they'd rather do those tomorrow, which means that we are wonderfully ahead of time, which means that we're going to move to the next session, which is drug discovery mechanisms and resistance. At the same time, the Geyser side meeting is going to take place, the closed session. So I think, actually, because all the dignitaries left, all of people in the back, I would like to recommend you move forward to fill up some of the spaces, to hear better, and also it looks better in the pictures. So if some people can move forward, and while you're doing that, the session chairs for the drug discovery mechanisms and resistance are Dr. Mrs. Evelyn Yahya Boni, a very good collaborator of mine, and also one of the founding members of WACCBIP, and Professor Samuel Dodu, who is the head of research training here at WACCBIP. Okay.
Thank you very much. So welcome to the next section, which is on drug discovery, mechanism, and resistance. So we have 4 talks here. So we'll do the first talk and the first 2 talks, then we'll have a break, and we'll come back and do the other 2. So the first speaker for today's, for this section is Jessley, our own. He's the postdoc at WACCBIP. And he's going to speak on differential susceptibility of Plasmodium falciparum to DHA and the static and suspension culture conditions. Okay. So Jessley, you have the floor.
Sorry. All right. Thanks. Yeah, that way. So, thank you. Good evening, everyone. Yes, my name is Jessley, and I'm a postdoc here at WACCBIP. So this evening, I'm happy to share with you data from my research work which seeks to study the differential susceptibility of Plasmodium falciparum parasites to dihydroartemisinin under static and suspension culture conditions. So, by way of introduction, Plasmodium parasites in the human body develop under different varying oxygen and nutrient levels. And because the parasites live in the blood, they experience shear stress from moving fluid. And this has been shown by numerous studies to impact their ability to invade red blood cells and also their growth and development. In contrast, the parasites that we use in the human in the lab settings for our experiments are grown under constant. They are grown in a static incubator under constant nutrient supply and temperature conditions, and they do not experience shear stress. So what this means is that static cultures, even though they may simplify experimentations in the lab, they may not fully reflect what occurs in the human body, and therefore drug efficacy and then resistance studies will require careful validation to ensure that these differences that occur within the in vivo settings and then the in vitro settings are taken into consideration. And this is important because understanding the environmental impact of on the growth and development of Plasmodium falciparum parasites is important in enhancing the translational relevance of whatever we discover in the lab. Right, this way. So, in line with this, a former colleague in the lab, Prince Nyako working with Prof, were able to show that using DD2 and W2 MEF parasites, they were able to show that these parasites grown in suspension culture conditions had the ability to switch from using salic acid dependent pathways to salic acid independent invasion pathways. And this also resulted in an increased expression of invasion related genes. What was interesting from their work was the fact that the Cal13 gene, which is associated with artemisinin resistance, was highly expressed in only the DD2 parasites, but not the DD2 parasites in suspension culture, but not the static cultures or any of the W2 MEF parasites, which was rather quite interesting, considering the fact that he was not interested in drug resistance studies. So I also found from his data that the multi-drug resistance gene that is associated with chloroquine resistance, as well as the AP2-ESP gene that has been known to regulate or modulate multiclonal gene families that are associated with host immune invasion and then adaptations to adverse environmental conditions, were also highly expressed in just the DD2 parasites, but not the static cultures or any of the W2 MEF parasites. So the question then was it possible that for the fact when we have parasites in the body, do they have the intrinsic ability to develop drug resistance or become susceptible to drugs as a result of just moving within the blood? And would this be mediated by epigenetic mechanisms? And so in line with this, I picked some of these parasites, the DD2 and the W2 MEF parasites, in static and suspension culture, and then set up a growth inhibitory assay to understand their susceptibility to dihydroartemisinin. From the data, I found that the W2 MEF and DD2 parasites that were in suspension culture had increased susceptibility to DHA compared to the static parasites. And this was confirmed using a ring stage survival assay. And so following this, I decided to, So following this, I set up a study to elucidate how epigenetic modifications in Plasmodium falciparum parasites may influence their sensitivity to dihydroartemisinin under static and suspension culture conditions, with the first objective to evaluate how susceptibility evolves over time especially under static and suspension culture conditions, using DD2 and W2 parasites to DHA. And following this, to use chromatin immunoprecipitation, couple sequencing techniques to identify markers that may represent epigenetic signatures of DHA resistance, and that may mediate epigenetic signatures that may mediate DHA resistance in the static and suspension cultures of DD2 and W2 parasites. And then subsequently to use RNA sequencing techniques to identify differentially expressed genes under static and suspension culture condition of the two parasites. And then finally to functionally validate these genes that we find to be mediating drug resistance. So for the first objective, the setup has mostly been the use of ring stage survival assay. So in this assay, we usually use highly synchronized ring stage parasites, and then we expose them to 700 nanomolar of dihydroartemisinin, and then 0.1% DMSO as a control. And then for my setup, I included artesunate at 3 times the concentration that we use for DHA to see if we could see a similar pattern. So after incubating the parasites with the compounds for 6 hours, we wash off the drug, and then we grow them in drug-free media for 66 hours, bringing the total time to 72 hours. So after which we use the flow cytometer to estimate the fluorescence levels or the parastemia, and then we estimate the survival, percentage survival of the parasites. So for this, the first, it's gone too far. Okay. So for the first part of the work, I used 3 laboratory strains, 3D7, DD2, and W2 as Prince had previously used in his work. And these parasite strains have different drug susceptibility phenotypes. 3D7 is mostly a drug-sensitive strain against most chloroquines, orthodoxin, mefloquine. And then DD2 and W2 are multi-drug resistant strains, parasite strains. So after 2 weeks of growing the parasites in static and suspension culture conditions, I observed that 3D7 parasites and DD2 parasites in suspension culture had increased survival compared to those in static culture conditions. But the contrary was true for W2 However, after 6 weeks, I found that the suspension cultures of 3D7 and DD2 parasite had increased, decreased survival compared to those in static conditions. And the contrary was again true for the W2 So it looks like there's a flip in terms of phenotype after 6 weeks. So following this, I picked some of the parasites that were in suspension culture and then put them in static conditions to see if there will be a recovery of the phenotype. So after 12 weeks, I found that for 3D7 and W2 parasites, the parasites that were in suspension culture that we had put in static conditions had almost similar survival levels as the static cultures, but you could see that the suspension culture were still had decreased survival. But this was not the case for DD2. We see that the parasites that were in suspension culture that we had put back in static culture were not able to recover, and they were actually they had less survival. They had a decreased survival advantage compared to the static and then those that were in suspension culture. And this pattern was very similar to what we observed with artesunate. And so following this, based on this data, although not all the data is, in terms of the comparisons were statistical significance, we want to repeat this work, but the data seems to suggest that it appears these parasites, there's a difference in terms of susceptibility or resistance between parasites that are grown in static and suspension culture conditions. And this changes with respect to the strain and also with respect to time. And so the idea, and this could be mediated by environmental factors. And so based on this, the next stage is to expand this process and then repeat the assays, and then also set up, detect epigenetic changes that may be mediating these differences that we are seeing. For the past 2 years, we've been working on a protocol to optimize a protocol for conducting ChIP-seq here at WACCBIP that would help us to detect epigenetic changes in Plasmodium falciparum parasites. And so we've been working closely with Prof. Rubin at the University of Edinburgh and his postdoc, Manu. And then here at WACCBIP, Joyce, and then Kukwa. So far, we've been able to optimize conditions for how much pellet material or parasite material will give us a good libraries in the end, or good concentration of DNA in the end. And then also in terms of conditions, we've optimized conditions for sonication. The antibodies that are used for the immune precipitation are those that are used in Robin's lab, and so we didn't really have to work on that. But we've also been able to optimize conditions for library preparation, and then we've sequenced some of the samples, and then we are trying to develop a pipeline for analyzing the ChIP-seq data. Yeah, so just a casual look at the data. So far for the optimization, we've used DD2 and 3D7 DD2 samples, and we had good concentration of libraries that had enrichment in target genes. So conducting a ChIP qPCR to detect, to see whether we have enrichment in the targets that we'll be looking for. We use primers against the AP2 gene that is shown to mediate sexual commitment in Plasmodium falciparum. So comparing that to actin, which is a control, we see enrichment in the target and the AP2 gene in the samples. And I've also been able to, after sonication and cleanup, we've had libraries that are within the right fragment length, between 200 and 700 base pairs, which are a requirement for the Illumina sequencing. So these samples have so far been sequenced. And I'm working closely with my colleague Collins and then Vincent, who manages the HPC. And I'm following a tutorial on the UCLA website to analyze, to see if I can develop a pipeline for the data. And so far, I've done alignment, peak calling. And so we have bed files and wiggle files that we can view the data using the integrated genome browser. And then we can also do other downstream analysis on the data. So this is just a casual look. This is one of the sample 3D7, and we can see some area that seems to show some kind of arrangement, peak areas in the data. We're still working on the data. So there's also analysis from the HOMA software that's used for identifying differential peaks. And for the 3D7, we see that it discovered 19 peaks that were that were enriched, or differential peaks, 380 differential peaks. Okay. So, so to summarize, I'd like to say that the current data that we have seems to suggest differences in susceptibility in also survival of P. falciparum parasites in suspension and static cultures of dihydroartemisinin and tisonate. And these variations would vary with respect to the strain and then the time. And what this means is that it's possible that parasites in the body would naturally be able to develop resistance or susceptibility to drugs without being exposed to the antimalarials, such that subsequently, when these parasites encounter a drug, they may become either susceptible or resistant to the drugs. So we are currently hoping to see what epigenetic mechanisms mediate this. And so with our optimized ChIP-seq protocol, we are hoping to elucidate the underlying mechanisms that is driving these differential responses to culture conditions, and what will be the implications of this for drug susceptibility testing. Okay, so to conclude, I would like to thank Professor Gordon Ondari, who is my mentor for my project, and then Robin that we work closely with, and then Kek here, who's also been very supportive in terms of the development of the CHIPSIC protocol, Dr. Niwe that I work directly with, and then I've already Daniel that I work with also the project, and then Joe, who started the optimization of the CHIPSIC protocol, and then I've already mentioned Joyce and Kukwa, and then Collins, and then Manu. Right. Thank you. I'd like to thank.
So we have 5 minutes for questions. So we'll take the first question from Prof. Kwashi.
Okay.
Here.
Yes, thank you so much. But I'm getting a little bit confused.
Okay.
Because I've been, do I say a culture of Plasmodium falciparum for well over 20 years, almost about 30 years. And the observation is that if we want a shortcut of growing the parasites,
yes,
to use an experiment, we just shake them and they grow.
Yeah.
So we have rather been trying to find out the mechanism why they grow faster. Some have proposed that the new permeation pathway, which occurs 18 hours after the parasite invade the red cells, the new permeation pathway is rather brought much or occurs much earlier.
Earlier, yeah. Okay.
It occurs about 12 hours instead of the between 15 and 18 hours.
Okay.
So what you are now bringing up is a little bit of worry, brings a confusion to some of us.
Okay.
So I want to believe, I don't want to believe that it has anything to do with the drug.
Yeah.
What it has to do is that some mechanism is brought forward, and so the parasite become very active earlier. It's just like a teenage pregnancy.
Okay.
So in that state, they naturally will be open to the effect of the drug. So unless you have some other mechanism to show me, you can do all the other things, but it boils down to the fact that shaking increases the activity of the parasites earlier. They begin their sexual activity earlier than usual. Thank you.
Yes, so that is indeed true. And from some of the papers that I've reviewed, they had actually looked at that the shaking, so the shaking actually increases the parasitemia, and most of that is regulated by epigenetic mechanisms actually. But what was interesting was the fact that, I mean, from Prince's work, I just saw that the calcitetin gene was up there, and it was only one that was very visible until I looked through the data to see what other drug-resistant genes were upregulated in the parasites that were in suspension culture compared to those that were in static culture conditions. But it was interesting that it was the multi-drug resistant genes they also had increased expression in only the suspension parasites of the DD2, not a static, and not any of either the W2MEF static or suspend culture. So what could be the mechanism? Could it mean that? And it was interesting to see that instead of being resistant, of course, if you, we know that this genes, this particular proteins are transporters, and so they are involved in the movement of substances in and out of the cell, which may be important for the growth of cell. And mutations in these genes are a state of resistance. So, it was interesting to see just only the DD2 parasite. So could this be a unique mechanism? I mean, could this be something that is just uniquely to only DD2 parasites? And if it is, what would be the implications in terms of drugs, which was why I went ahead to look at, okay, the calcitonin, expression of the calcitonin gene is up, but does it have a corresponding effect on its drug susceptibility? And we see that if you compare the static and dose in suspension, it had decreased susceptibility. But if you are comparing the DD2 and the W2MEF, you see that the W2MEF had increased susceptibility compared to that of the DD2 parasites. So there's something interesting happening here that we cannot fully pinpoint at the moment that this is the exact thing that is happening. But considering the fact that the shaking creates that kind of epigenetic environment, we're thinking that there could be some genes, this transporter could be epigenetically regulated. And so, which is why I want to go downstream to see what are the underlying mechanisms that is defining these differences in terms of the phenotype between the different strains. But it's possible that this mechanism we are just seeing here could be actually happening in the body. And that's what I'm just thinking that.
All right.
Yes.
So just, I think it's fine.
Okay.
Let's take other questions then.
Okay. Thank you.
Yes, please.
Thank you very much. Very talk. I'm trying to really connect the dots here because you start off your talk by really painting a very relevant application of what you're trying to do by emphasizing how the parasite in the body will be in different conditions and why in the lab a similar growth condition are used to test drugs. But towards the end of your talk, I don't know how the chip that you are developing would translate into application. Again, remember that most of what you are describing might be transient phenomenon. And if your aim is to develop a chip that will type this different variant, is it to use it directly to type a parasite living in the blood, or the parasite had to go through a growth process before you type them? Because, I mean, that growth of the parasite actually lead to selection of some of the things that you end up seeing, which might be very different from what come from the body. So I think my question is really trying to connect where you start up with by acknowledging the differences that you find in parasite stature in different human body to what you actually see in the lab. And I think I'm still missing how are you connecting the two?
Okay, so if I get your question correctly, you're thinking about what the parasites in the body as opposed to those that were used in the lab. Is that okay? Yeah. So we know. So to
put it simple, I mean the chip that you are developing.
Yes.
Is it to type resistance of the parasite that are living in the body?
So the chip that we are developing, the chip is actually chromatin immunoprecipitation coupled sequencing. So it's a technique that is used to detect epigenetic modifications in any organism. Yeah. And so we are using this technique to understand the epigenetic modifications that are arising from the different environments that these parasites are growing in, that is static and suspended parasite conditions. So we'll have parasites. So at this time point, we are looking at maybe after 2 weeks, keep one in suspension, one in static, and see what is. We know there are differences from previous work, on innovation assay, but now want to know what epigenetic modification is driving this difference that we are seeing in the two conditions, and then we can connect that to what is happening in vivo.
Okay, thank you.
All right.
All right. So we will last question.
Thank you very much. I think picking from this question, are we saying that you're talking about the parasites in the human body, that is in vivo parasites?
Yes.
Then the parasite that is being used in the lab, in vitro parasites.
Yes.
Now, from the start of the talk, it was as if we want to do a comparative analysis to see the difference between the parasites in vitro and the parasites in vivo. But the work is still parasites in vitro. But one is suspended, while one static, while the other one is not completely static. So at what point or how are we mimicking the parasites in vivo?
Okay.
I don't know whether you get...
Yeah, I get
thank
that.
you.
So we're not fully mimicking the in vivo culture conditions, but we know the parasite in the blood is in suspension culture. So it's not static. It's not, although there's sequestration, there's also sequestration of the parasite. So then they are found in a certain compartment. But if the parasite is in the blood, it's in suspension and it's moving against the fluid and all of that. And so the shaking in the lab is supposed to mimic that environment, yes, but it doesn't fully take account of what is going in vivo. So I'm sorry that didn't come up clear.
All right. So thank you so much, Jessley, for a great presentation.
All right. Thank you.
Thank you, Jesse. We'll have the second presentation by Serge Mostowi. Yes. He'll be speaking on the use of cytoskeleton to control Shigella infection. You have 15 minutes for your presentation, please. Thank you.
If we could dim the lights, that would be in the front. Is that okay? Thank you very much. It's a great pleasure to be here and to try and contribute to this very high level of science. I thank Gordon very much and the other organizers for considering me to present my work. So I'm Serge, I'm based at the London School, and I want to tell you about my lab's efforts where we try and use the cytoskeleton to control Shigella infection. So we're a cell biology of bacterial infection lab, and we actually use a variety of pathogens. In particular, we like those that invade normally non-phagocytic cells. They escape from the vacuole, and they polymerize these absolutely wonderful actin tails. So it's bacteria that hijack the host actin cytoskeleton to polymerize these tails and to disseminate from cell to cell. And in that way, they spread across tissues, and that process is absolutely essential to cause disease in humans. In my lab, we focus on the gram-negative bacterium Shigella flexneri. First and foremost, it's a very important human pathogen. Hundreds of millions of illness episodes per annum, hundreds of thousands of deaths, particularly in children under 5. I must say 2025 is a bit of an electric time to study Shigella. The WHO has highlighted as the highest priority pathogen due to its ability to acquire antimicrobial resistance, and there's no licensed vaccine. So there's great attention right now to study Shigella. In the lab, we like Shigella because it's closely related to E. coli. In fact, it's taxonomically indistinguishable. It's 99.9% the same. The big difference is it's got a virulence plasmid, and on that virulence plasmid is encoded a molecular syringe. It's a type 3 secretion system that injects itself into the cell to really spew out these effectors to manipulate different aspects of the infection process. So as a result, for the past 30 plus years, Shigella has been a fantastic discovery tool in both cell biology and innate immunity. In fact, if you think of some of the important innate immune defenses discovered in our lifetime, bacterial autophagy, nod-like receptors which underlie Crohn's disease, neutrophil extracellular traps, guanylate binding proteins, Shigella flexneri has been ideally has been there as if not the bacterial pathogen to discover that process. I can see here, should I continue? No, please. Boom. Wonderful. And so in my lab, we study cytoskeleton rearrangements. Most labs study actin or microtubules. We study an atypical component of the cytoskeleton referred to as the septins. So septins were discovered by Nobel laureate Leland Hartwell. He shared the Nobel Prize with Sir Tim Hunt and Sir Paul Nurse. And they're essential for cell division. So it was discovered in budding yeast, but we know that septins are essential for all forms of cell division. As a result, the majority of the field studies the role of septin biology in dividing cells. In humans, it's a little bit complicated because we've got 13 different septin proteins that assemble together to form heterooligomeric complexes and filaments. So this jelly bean cartoon depicts how we need one member of each of the different septin families to form these complexes. These complexes will form filaments that can then superassemble into bundles or these ring-like structures. What's important is that septins can associate with actin, microtubules, and membrane. However, because they were discovered for their roles in cell division, very little is known about their roles in non-dividing cells. In order to understand their role in non-dividing cells, we infected them. And really that's the premise of cellular microbiology. We wanted to learn about septin function during the infection process. So I spent many years trying to understand what septins were doing at the phagocytic cup. So we knew these actin-rich processes were essential to enable Shigella internalization. And we discovered how these septin ring-like structures were required to promote internalization. I spent many years but did quite a poor job. I still don't really know what septins are doing at the base of the phagocytic cup. But the one key experiment we did is we treated cells with cytochalasin D. So it's a drug to inhibit actin polymerization. And in that way, septin rings no longer assembled and they no longer recognized bacterial entry. However, at the time, there was another actin-based process, and that's actin-based polymerization. Those are the actin tails I referred to. And so we wondered if septins would respond to actin polymerization in that way. And you can argue that was probably my greatest hit as a postdoc. We discovered how these septin cage-like structures will require actin polymerizing bacteria in the cytosol and compartmentalize them for degradation. So that was the first time the cytoskeleton was linked to innate immunity. So it was the first time someone discovered that you could stop actin-based motility, and septins were the first cytoskeleton protein as involved in cell-autonomous immunity. It was actually based on that discovery I wrote to the Wellcome Trust. I said, I want to start my lab on this process, and I want to see if I can have human health impact. So this is the type of movie we would see at the time. So it's a HeLa cell, a human epithelial cell, transfected in green for actin and in red for the septin cytoskeleton. And you can focus on the merge. It's a 90-minute movie sped up for your viewing pleasure, but it'll loop. And so the first thing I hope you appreciate are these absolutely wonderful actin tails. And these are how bacteria are running away from cell autonomous unity. They're avoiding autophagy, they're avoiding ubiquitination, and they're spreading from cell to cell. It's these two bacterium in the top left corner you'll notice. So bacteria are presumably still alive because they're trying to polymerize actin, but they're compartmentalizing these red cage-like structures. Presumably the septin rings are preventing the polarity of the actin tails and keeping it in place. So we spent many years doing functional work to show that if you increase septin expression, you dramatically reduce actin tails and you completely prevent dissemination. Whereas if you deplete septin expression, you get more tails and more dissemination. So it was a bona fide component of host defense. Unfortunately, at the time, the glamour journals weren't completely satisfied with that because they wondered what was the fate of bacteria inside of these cage-like structures. And so those were some of the first questions we would ask. Single-cell bacterial cell biology, I would argue, is still very difficult to know if these bacteria are living or dying. And so at the time we were able to link it to bacterial autophagy. It was an important time. Felix Rando at the LMB or John Vermel at the Sick Kids in Toronto were discovered that something that was ubiquitinated about the bacteria or maybe membrane around the bacteria were actively targeting bacteria to degradation by autophagy, the intracytosolic defense pathway. And so what you'll appreciate here are two hallmark autophagy markers. This is the autophagy receptor P62, and this is LC3. That's what lipidates into the double membrane. And so something about the bacteria will be ubiquitinated, P62 will recognize that, and then LC3 will build the double membrane to autophagy. And so from this slide, you can see how septin cages were associated with targeting bacteria to degradation by autophagy, really solidifying their role, their antibacterial role. So for us, it was key to understand how are septins recognizing bacteria in the first place. And this was really done by Sina in the lab, who went on to get an MRC Wellcome Postdoc Award. So this was a true eureka moment. And so we were able to get a microscope that was fast enough to capture septin cage entrapment in real time. And so these are just stills from this movie. And what I hope you appreciate from this movie is that the first instance of septin recruitment is to the site of bacterial cell division. And so this was a really romantic concept for us, because I told you that septins were discovered as being essential for host cell division. In 2016, we were able to show how septins were able to recognize dividing mitochondria. And considering that mitochondria are viewed as ancient bacteria, it was really quite a stunning observation that there was something about dividing bacterial cells that septins would recognize. And so we needed to understand the underlying mechanisms. Going through about 4 years of data, it turns out that septins are really a biophysical determinant of host defense. So in a normal case, you have Shigella, it would actively invade cells. They express anionic phospholipids at their poles, and they have something called FCFZ. It's a tubulin-like structure that will invaginate to form a septum. These bacteria will then go on to divide, form actin tails, and cause disease. So what's remarkable is that we have a host cell system that recognizes actively dividing bacterial cells based on their size and shape. Septins were described as micron-scale sensors of curvature. So they recognize curvature on the order of 1 micron. And in that way, we were really quite lucky, because Shigella is 1 micron in diameter, 3 microns in length. And so what we discovered is that septins recognize the micron-scale curvature presented at the poles of bacterial cells. They interact with the anionic phospholipids. For reasons we don't understand, as the bacteria divide, they have to grow a little bit, and that really kind of enables the full septin cage entrapment, and then that's a death sentence for the bacteria. We know that 93% of the bacteria will not be allowed to escape from these cages. They will then be targeted to degradation by autophagy, really highlighting the antibacterial role of septin cage entrapment. So that's the working model in the lab. We need to understand if septins on their own are antibacterial. Really, all of our work has shown that it somehow coordinates its antibacterial activity with autophagy. So Gizem is a Human Frontiers funded postdoc in my lab. She's actually a biophysicist by training. And so she came into the lab to understand how septins could interact with different bacteria of sizes and shape. And so to enable this project, we developed a purified protein system. We need to study outside of cells where there's too much complexity. And so we were able to purify septin cages in isolation. So in the absence of any other host cell factors, we could generate septin cage-like structures around a subset of our Shigella cells. And so these are purified septin proteins labeled with GFP. And what I hope you can appreciate is that the cages look to be quite faithful to what we see in cells. We see these ring-like structures. And so now we have a very efficient screening platform to understand what are the factors that promote or prevent septin cage entrapment. And so the first thing Gizem did is we could assess what was the antibacterial of septin filaments on their own. And so for that, we could treat cells with or without purified septins and assess their growth. And so what is immediately striking is that in the absence of septin, in the presence of septins that can form different type of heteroalgomeric complexes, we had a dramatic reduction in length. So roughly 1/5 of the bacteria failed to grow. The bacteria grew 20% less in the presence of these septin proteins. This just shows that you need filament formation for maximum restriction. So this was an important slide for my lab because it shows that septins possess intrinsic antibacterial properties in the absence of any other host factors. So how are septin filaments, as they interact with the bacterial surface, on their own antibacterial? So because of our system, we could actually interrogate the bacteria and ask how they felt about septin cage entrapment because they're completely isolated. And so we could perform RNA-seq on bacteria that were entrapped in cages or not. And the profile that we obtained was really remarkably consistent. And so we had a very tight list of genes that were upregulated in the presence of septin cage entrapment. First and foremost, that molecular syringe I introduced at the beginning, there was something about Shigella that senses septin binding as a danger signal, and in that way it goes up. The other thing we noticed was stress response, and it was a stress response induction that was tightly linked to really alterations in membrane polarity. So we could go on to test that in time. I don't really have to show, but really there are two striking messages from this manuscript in preparation. One, bacteria can somehow recognize and tolerate septin binding. So it's a new way to think about tolerance to cell autonomous immunity. And 2, we show that septin filaments on their own, they don't cause damage to bacterial membrane. What they do is they induce differences in membrane polarity, and in that way affects bacterial cell growth. Septins bind a wide variety of membranes, including neurons, so the concept of septin membrane interactions having impact on polarity has very far-reaching consequence. So all of that work was performed in vitro, and in order to understand how septin cage entrapment is coordinated with other aspects of cell autonomous immunity, Anna was a Marie Curie-funded postdoc in the lab from Switzerland who wanted to understand what are the other factors that recognize Shigella during the infection process. And so now we've gone back to HeLa cells. And so when we infected HeLa cells, we performed a proximity biotinylation approach to try and understand all of the host and bacterial factors that were within 10 nanometers of the Shigella surface during the infection process. So this is a very innovative approach because we could functionalize the bacterial cell surface. So we had Shigella express these apex nanobodies, and then we perform proximity biotinylation. You can see the streptavidine label really indicating the peptides that were being recruited to the Shigella surface. We're then able to send these to Francis in Ghent, who performed very high quality mass spec to identify host and bacterial peptides that were recruited to the Shigella surface. The paper has recently been posted on bioRxiv, but this was really a transformative approach to understand how cages are coordinated with the rest of cell autonomous immunity factors. And in this paper, we highlighted a novel E3 ligase that was recruited to the Shigella cell surface. Another way in which we're studying septin cage entrapment is we're performing high content, high resolution microscopy. So we really have a unique unit of cell autonomous immunity, the Shigella septin cage. And so we want to discover pharmacological compounds that would promote or prohibit this process. So I was lucky enough to get an ERC consolidator grant in which we were able to purchase this high content, super resolution microscope. We needed that additional resolution to visualize the filaments wrapped around bacteria. The first thing you'll notice is that this microscope actually doesn't have any oculars, so it doesn't require a human anymore. So we required artificial intelligence and employed convolutional neural networks so that the microscope could automatically identify septin cages in each of these 96 wells. So here's an example of positive recruitment, negative recruitment, and intermediate. And in this way, it's really kind of revolutionized septin cage biology in our lab because it no longer requires human interpretation. You know, we can now have a microscope dictating to us what is positive or not, and we are now really well suited to employ high content screens for different pharmacological compounds. For my final slide, we need an animal model if you ever want to have human health impact, and unfortunately Shigella actually doesn't work in a mouse. So there's something about the intestinal lining of mice that just, you can pump a mouse full of 50,000 bacteria and the Shigella will pass right through. On the other hand, in the case of humans, it takes less than 10 bacteria to make us sick. So there are humanized mouse that are susceptible to Shigella, but I hope you can appreciate for our cell biology questions mice have fur. And so instead of mice, we turn to the zebrafish model because when embryos are born, they're optically accessible. And when they're less than 5 days old, they're not considered animals by law. And so we can tag any cell type we want in a color, we can infect with our fluorescent bacteria, and we can watch the interaction of Shigella with stem cells, macrophages, neutrophils in real time at high resolution. So it's been a transformative process to understand the Shigella infection process in vivo. And so these are just the different developmental stages of our zebrafish larva. And we've used the zebrafish model in a variety of different ways. We use it to understand the cell biology of bacterial infection. We use it to understand how Shigella can interact with macrophages and neutrophils. We've used it to study clinical isolates. So for most of today, I've talked about lab strains of Shigella that have really enabled all this immune advance. But of course, there's really quite a significant variation from clinical strain to clinical strain. And so we're employing our zebrafish model to understand those genetic differences. We've developed it to understand how host responds to bacterial interactions. And because there's no vaccine for Shigella, we're trying to develop or try to harness training innate immunity to come up with a better Shigella vaccine. I'm going to end there. I'm really happy to take any questions and to discuss throughout the meeting. It's a real pleasure to be here. I mentioned the key people throughout the talk. I thank the key funders, and I really look forward to interacting for the rest of the meeting. Thanks very much.
Thank you, Serge. Please, any questions? I don't see. Okay. 2. 1 here.
So if we've evolved this septin cage to kill these bacteria, how come we all get sick with only 10 bacteria?
Thank you for your question. So it turns out that Shigella has evolved different effectors to block septin cage entrapment. So the process that I've described for you today recognizes a fraction of bacterial cells on the order of 15 to 20%. Last year, what was discovered, so there's a group in China that for the first time discovered, so Shigella has the type 3 secretions. Upon that, it secretes an effector. And so we now know that there are 2 effectors that are dedicated to blocking septin cage entrapment. One modifies the ubiquitination machinery to prevent septin filament assembly, and there's a new paper under review in which there's a different effector that directly targets one of the septins to prevent its assembly into cages. And so I hope people can appreciate that Shigella has evolved 2 different ways to block my mechanism of host events. Kind of gives me street cred because it must be really important for the Shigella infection process.
Do you know how it translates to other bacteria?
So thank you for your question. So we really use Shigella as a model. As I highlighted, it's kind of a landmark tool for innate immune discovery. We know that septins can recognize a wide variety of bacteria, for example, Staphylococcus, that's gram-positive, Mycobacteria marinum, which is the fish version of tuberculosis. So septins are quite promiscuous in the type of lipids they interact with, but the shape of the bacterial cell really seems to matter. So if bacteria are too big or too small, that may be a weakness of septin cage entrapment, but it is of great importance to us to understand the breadth of pathogens that septins can recognize and modify it so it can recognize all of them.
Is there any deficiency of septins in human which may make more susceptible to infections?
So that's an absolutely wonderful question. So I grew up in the Center of Host Resistance at McGill University. That was my PhD. And I was always exposed to what are the genetic determinants that predispose humans to infection. So I've looked through the databases of Jean-Laurent Casanova, who's at Rockefeller, who has all these immune deficient patients. There are, mutations, but there's a lot of work to be done to directly link them as the causative agent. So it would absolutely be, it's definitely something I would like to look for, but I have no evidence that there's direct mutations. I want to highlight that there's no such thing as a human born without septins. Cells would fail to divide. So you could really only have slight mutations. And so I think what's very fascinating is that septins are deeply involved in mental health and all kinds of neurological disorders. So I haven't really seen, some of them are often triggered by infection. But as to whether or not mutations in septins predispose to infection has not yet been linked, but I'm looking for it. It's a very high bar, by the way. I hope you don't think less of me. It's a very high bar. If you think of a lot of innate immune things, TLRs, GVPs, autophagy, have they been linked to infection? It's always not so clear.
I'm trying to...
That's a very deep question, because I often get criticized that Shigella is a human-adapted pathogen. So Serge, why on earth are you infecting zebrafish, which is so far away? So what I would argue is that I am really studying very fundamental concepts of host defense, very fundamental aspects of innate immunity. And the beauty of the zebrafish model is you can dilute drugs in the bathwater. So I've got 96 well plates where you can put different concentrations of any pharmacological compound you want, and the fundamental concepts of Shigella infection control. Fundamental concepts of Shigella infection control.
Thank you very much. Please, do you have any questions online? No, any comments online? No? Okay. Please, we have to take a break now.
Okay. Thank you.
All right. After that session, we are going to take a 15-minute break. I'm stressing on 15 minutes so that we can still stay on top of time as we are right now. And to our moderators, please, you are still handling the session after break. So please, the ushers would take you to where we had the morning coffee break. For a quick refresher, and please come back. When you return, please start sitting from the front instead of the back. Thank you so much. 15 minutes. 4:35. 4:35.
Hello?
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region, because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the ring. So I called Gordon and said, Gordon, I think you should host this.
So basically I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Theresa, Osborne, Abida. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention. I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than $50 million for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honor for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the number one leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACCBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACCBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the great continents.
WACCBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACCBIP has more than prepared me, and I wouldn't say this because I've just stayed in WACCBIP, but for the fact that WACCBIP has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WACCBIP was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WACCBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WACCBIP did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WACCBIP did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, You don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WACCBIP machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork and very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WACCBIP because the leadership of WACCBIP identifies young talent and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, schools engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WACCBIP has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WACCBIP has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies.
Hello, how are you doing today?
Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients that we need a sample of her blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Doc, please, we are here.
Oh, good. I hope you are well. Don't worry. We are here to help you, all of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay, for our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? We need a sign language interpreter.
Hey, stop looking at them. Who told you I'm interested? I can see it in your eyes. See. Doris, they are deaf and dumb people. It's true. Ah, I hear these people are cares though. How do you even introduce them as your friends?
You know, every physical condition has its medical cause. It has nothing to do with curses.
Session. We have two key speakers here for this section. The first, or the third speaker, is Mami Ekwia Akwa, and she's going to talk about an optimized bacterial and insect expression system to model human ACE2 and Saskov-2 interactions. Mami Ekwia, you have the floor. All right.
Thank you. Fastest presentation ever. Okay, so good afternoon, everybody. My name is Mami Akwa, and today I'll be sharing with you parts of my PhD research work on an optimized bacterial and insect expression system to model human ACE2 and SARS-CoV-2 interactions. Okay. Okay, so in 2020, we were hit with the COVID-19 pandemic, and a few years down the line, in May 2023, the WHO declared that the pandemic was over. Since then, we've had globally over 700 million cases with more than 7 million deaths. In Ghana, we recorded about 172,000 cases with more than 1,500 deaths. Recently, there has been an increase in COVID-19 cases reported, and even recently, an outbreak on the University of Ghana campus. Now, we know that with all these outbreaks, there are different variants that have dominated some waves of the pandemic, and even more recently, we have variants such as the JN1, which emerged in 2023, the LB1, which is a direct lineage from that, which emerged last year, and this year we have the NB181. Now, even though we're able to mount good interventions during the pandemic, we still do not understand asymptomatic infections and host viral dynamics. So it will be important to address these towards future pandemic preparedness. So with the gap with host viral dynamics, It is crucial to understand how the virus binds to the host via the angiotensin converting enzyme 2, and this is what drives SARS-CoV-2 pathogenesis. Additionally, it has been shown that different variants of SARS-CoV-2, specifically the RBD, could increase binding to ACE2, and different polymorphisms in ACE2 could increase or decrease binding. So if we are able to understand this interaction, we could possibly have insights on binding mechanisms, severity of infections, and understand the asymptomatic narrative. So the approach I'm choosing is to use a bacterial cell culture via the E. coli expression system to express the RBD of SARS-CoV-2, and then to use insect cell culture via the baculovirus expression system to express the human gene, which is the ACE2, put these two expressed proteins in an interaction via ELISA, and then look at the ODs and binding affinities, with the aim of developing an in vitro system to model what host viral interactions look like. So first, I began with an in silico interaction assay, looking at the viral RBD and the host gene, and I used Prodigy to calculate the binding affinities. Now, what I observed was that ACE2 with Wuhan, which is the wild type, and KP311, which is one of the newly emerged strains, had very high binding affinity as compared to Omicron and JN1. However, for Omicron, it has been shown to be a variant that is dominant in immune escape, and JN1 also has this ability. Now, considering that KP311 evolved from JN1, it is understandable that KP311 has a higher binding affinity as compared to JN1. So after doing the in silico, I moved on to the in vitro steps. So I have to mention here that this part of the work was done at the European Molecular Biology Laboratories in Grenoble, France. So I did construct and plasmid design with an in-house vector, put my ACE2 N-terminal peptidase domain into this vector using the baculovirus cell culture. And then I did the same for the RBD with another in-house vector, and I used bacterial cell culture for this. And for the RBD, I specifically looked at 5 variants, that is the Wuhan, Omicron, JN1, KP3, and LB17. So for the bacterial cell culture, after successful transformation, I did a pre-culture, a large-scale culture, and then I monitored my OD to about 0.6, added IPCG to induce protein expression, and I harvested the cells. On the other hand, with the host gene, I used the insect cell culture. So after also successfully confirming transformation, I put my gene of interest into a transfer vector and transformed that into the DH10 E. coli cells, which contains the baculovirus genome. I did a selection with blue-white screening, and then selected white colonies, which now had both the baculovirus genome and my insert. And then I extracted the BACMID from this, transfected SF21 cells, went ahead to do a V1 amplification, and finally a large-scale protein production in HI5 cells. So the next step was protein purification, which I did after cell lysis and filtering my lysate. I used difference buffers, ran it through a nickel column, and collected fractions at each stage, which I ran on an SDS page. So for the SDS page, for the RBD, I was expecting a protein size of 25 kilodaltons, which I saw here, but was very faint. So I went ahead to concentrate the protein, and I got a higher concentration. On the other hand, for the ACE2, I was expecting a band size of about 80 to 85 kilodaltons, which I got in my total fraction, but I missed in subsequent fractions. Now, since the goal was to use both host and virus proteins in the ELISA, I went ahead to get this kit, which had ACE2 coated on the plate. However, this is an inhibitor screening assay. So I was trying to repurpose it to look at the interaction between ACE2 and RBD. As the RBD2, I was able to successfully express 2 variants, which is the Wuhan and the LB. So this is my experimental setup for the ELISA. So I had 2 main concentrations for each variant, 15 nanometers and 20 nanometers. I had my controls, and then I had inhibition positive and inhibition negative controls. So analysis on this part is still ongoing, but this is what I have observed so far. So kindly follow the white boxes. So for the Wuhan, I got a low OD for 15 nanometers and a higher OD for 20 nanometers, which could possibly mean that the OD increases with decreased concentration and could possibly be because of aggregation with high concentration. Now for the LB, I got what I would call the expected trend, where binding increases with concentration. Now to my controls, which are a little tricky because I was repurposing an inhibitor assay for what I wanted to do. So for the positive control, which is an S protein, I had an OD of 0.8, and then the Wuhan with the positive inhibition control, I had an OD of 0.4. With LB and the positive inhibition control, I had an OD of 0.9. And then for the negative control, which was just the diluent, I had an OD of 0.9. And then the negative inhibition controls were 0.4 and 0.9 for Wuhan and LB respectively. Now, the S protein is the spike protein. So this tells us that even at low concentrations, the spike protein will bind to ACE2. However, we observe a positive inhibition of Wuhan and no inhibition of the LB17. Considering that the LB17 is an Omicron variant and has immune escape capabilities, this inhibition control was a neutralizing antibody. So this could explain why we still saw no inhibition in the case of LB. And then for the negative controls, we didn't see any significant effects on binding. However, with the diluents only, the high OD could be because there was still antibody detection. There was still the detection antibody in the well, so it could be non-specific binding. So with this, today, I have told you that it's important to understand host viral dynamics, looking at host genetics and viral receptor binding towards future pandemic preparedness. And what I'm proposing is that we use insect cell culture to express host proteins because it has the advantage of post-translational modifications. And then we do the viral protein by bacterial cell culture because they're usually smaller in size. We put this in an interaction assay, both in silico and in vivo, as well as in vitro, so that we can understand binding mechanisms, severity of infections, and symptom disparities. So with that, I'd like to acknowledge my supervisors, these funding organizations, and everyone here. Thank you.
Yes, so thank you very much, Mami Ekua. You have questions? We can take some questions. So I was just thinking about using the insect culture for the expression. You say, it allows post-translational modification.
Yes.
What about the quantity? Do you think you can get much more expressed proteins there?
Yes, so with the insect cell culture, because the large scale is almost comparable to the bacterial cell culture, so you're able to express a high volume of proteins.
Okay. Yes, there's a question.
Purification using the column.
Yes, please.
Were you able to have a purified band, because I saw multiple bands around? Then 2, regarding your ELISA, why did you choose 50 and 20? I was thinking there should have been a titer for you to know which is best. Perhaps that may explain why you're having your inability to know if the specificity or not? That's just, thank you.
Okay, so with regards to the purification, I have planned to do a dot blot assay to confirm that my proteins are the proteins I expect, even though I had multiple bands. And with respect to the ELISA, I did an optimization assay, and then I realized that these concentrations were the ones that worked best so far. Thank you.
All right, any more questions? Okay, no. Thank you very much, Mamekwa, for the presentation.
Thank you, Mamekwa. So, we have our last presentation for the session, which will be delivered by Tosin. E. coli persistence remodels gene expression to invade bacteria.
Good afternoon, everyone. Okay, good afternoon once again. My name is Tosin, and I'm here to give a presentation on a part of my PhD research titled E. coli persistence remodels gene expression to evade bactericidal antibiotics. I mean, that's just the fancy title for gene expression of persistent cells. So to start with, I'm going to introduce you to different bacterial phenotypes in response to antibiotic treatment. And here we have susceptible cells, which are usually killed by antibiotics at expected therapeutic concentrations. Then we have resistant cells. These are actually cells that carry genetic mutations and acquired resistance genes, and hence they are able to grow in the presence of antibiotics. So we also have persistence, which is a new mechanism of antibiotic resistance, and it's a phenotypic survival strategy which bacteria use to transiently enter a dormant or slow-growing state in order for them to survive high concentration of antibiotics without genetic resistance. So while persistence is at subpopulation level, we have tolerance, which is usually at population level. So this means that these two are sometimes used interchangeably. So for my study, I focused more on persistence. So although the mechanisms behind persistent formation in bacteria is still under investigation, I'm going to be showing on this slide some of the stress-driven processes that can contribute to the induction of persistence. So when you have a population of susceptible bacterial cells exposed to either antibiotics or environmental stressors, it triggers a series of internal responses, including transcription remodeling, which is the same as changes in gene expression, growth rate reduction, and sometimes accumulation of adaptive mutation, which then shifts a subpopulation of the cell into persistent phenotype. So while resistance is at genetic level, we have persistence, which is a reversible and non-genetic adaptation to stress. I'm sure some of you are wondering, why are bacterial persisters important? So this slide is going to summarize why these persisters are clinically significant. In an infected patient, we can the population of bacterial cells usually include the susceptible and also the persistent cells. So when antibiotics are administered, there will be clinical improvements. The person will probably feel better, and once the treatment stops, unfortunately, while the antibiotic treatment is ongoing, it will kill all the susceptible cells, and then we have the persistent cells that will survive because of their dormant like states. However, since there is clinical improvement and they have probably gotten to use the antibiotics following the prescription. Once the treatment stops, the persistent cells can then undergo cell division and repopulate into another population of susceptible pathogenic cells, and this cycle would continue. This usually results in recurrent infections like urinary tract infection, tuberculosis, typhoid fever, amongst others. And also it can contribute to treatment failure, biofilm infections, and sometimes promotes evolution of antibiotic resistance. So despite this, the clinical significance of persistence, there is scarcity of studies, especially on among clinical isolates, especially in Africa. Majority of studies, even outside, have used laboratory adapted strains to study persistence. So for my study, I'm going to summarize characteristics of the E. coli strains that are used for the persistence assay. So the E. coli isolates were obtained from 2 sources, the clinical from septicemia patients, and we also have the environmental isolates from ICU formats, such as doorknobs and surfaces. So using genomic analysis, we were able to identify the pathotypes of these strains, and the clinical isolates belong to the extraintestinal pathogenic E. coli, while the environmental isolates is uropathogenic E. coli. So we went on further to look at some of the genome features of these E. coli strains, and they have shared genome features in terms of their resistance and virulence factors, as well as mobile genetic elements, which means that they have the capability of transferring some of these virulence factors from one bacteria to the other. And phenotypically, these strains are resistant to multiple antibiotics, and they have strong and moderate biofilm formers, and they tolerated extreme stress conditions like high temperature, alkaline pH, and osmotic pressure. So then this led us to ask the following research questions: that can the E. coli strains actually tolerate and persist under bactericidal antibiotics, and what would be their gene expression profile? So our goal was to study the genetic basis of persistent formation in E. coli isolates obtained from tertiary hospitals in Ghana. And to achieve this, we study persistence phenotypically using these strains under antibiotic pressure, and then we used RNA sequencing to profile the gene expression pattern to compare between the E. coli isolates that are treated with antibiotics and control, in order to identify relevant genes and pathways that might be involved in persistence. So this is the experimental workflow. I used 2 clinical strain and 1 environmental strain, and I used the meropenem and ciprofloxacin for the antibiotics stress, and I also stressed them on that temperature at 45 degrees C. So after the antibiotic treatment, which was for 3 hours for the RNA sequencing, I washed centrifuge and extracted RNA for sequencing. Then I used bioinformatic analysis for the gene expression profile. So for the persistent formation assay, I used minimum inhibitory concentration times 2 of the minimum inhibitory concentration in order to rule out resistance effects, so that we can actually focus on the persistent cells. And this figure here, the A illustrates the optical density of the cells, while the B is showing the viable cell count after exposure to times 2 minimum inhibitory concentration of meropenem for the duration of 5 hours. So in the OD graph, we observe a sharp decline, which is consistent with rapid killing of susceptible population after introducing antibiotics. And then following this, we observe a low and stable decline over time This indicates that there's probably no significant cell division or regrowth upon exposure to antibiotics. And similar pattern is observed for the colony forming unit, where we also observed like a steep decline after 1 hour and then gradual decline over time. This plateau behavior is consistent with persistent, like hallmark of persistence, which means that a subpopulation of bacterial cell can actually survive without active replication. So we did similar thing using ciprofloxacin antibiotics. And in contrast to meropenem, although there was also initial decline after introducing ciprofloxacin, we observed that some of the strains were actually had increased OD values during the treatment period. And this is also consistent with the viable cell counts, the colony forming unit. So this means that although they were able to form persistence under ciprofloxacin antibiotics, persistence can actually be antibiotic dependent and also strain dependent. So, and also it's possible that some of these cells are, like, even though they are non-growing or slow growing, they were still able to maintain metabolic activity. So we went on further to see if prior exposure to heat stress can actually influence their survival under these antibiotics. So what we did was to prior exposure to temperature can actually prime the bacterial cell or possibly induce some stress adaptation pathway, which can increase their survival under antibiotic stress. So for the transcriptomic analysis, there are 3 different conditions. We sequenced ciprofloxacin persister cells, temperature recovered ciprofloxacin cells, and also the temperature tolerant cells. And untreated stationary phase cells were used as control, and that's because they are usually, bacteria during this phase are usually dormant or slow growing because of nutrient deprivation and other factors. So then using adjusted p-value less than 0.05 and log 2-fold change above 2, we were able to select upregulated genes and below 2 for downregulated genes. And this volcano plot is actually showing extensive gene expression profile under the ciprofloxacin treatments, and majority of the genes, differentially expressed genes, are above 500 for under all the conditions. So importantly, we used KEGG pathways to actually analyze the pathways in which these genes are associated with, and we can see here that majority of the genes are associated with metabolic pathways, indicating that these stress-adapted strains are probably using metabolic reprogramming for their survival. And this is consistent with a recent study published by Rahman et al., where they looked at laboratory-adapted E. coli strains, persistence forming ability of laboratory-adapted E. coli strains, and they were able to conclude that although they are non-growing cells, but they are still metabolically active with high number of genes being expressed. So we thought it would be interesting to see the top 60 differentially expressed genes, and also to help us appreciate the difference between the control, which are the natural dormant cells, and the persister cells. So if you look at this heat map, it's showing, you see like a reciprocal gene expression pattern where genes that are downregulated, the blue is downregulated and the red is upregulated. The genes that are downregulated in the control cells are actually upregulated in the persister cells, and vice versa. And you notice that the middle parts, which are the temperature tolerant cells, also have some type of unique expression patterns. So to understand the biological processes in which these genes are involved in, we grouped them based on function, and the dominant biological pathways include biofilm regulation, metabolism, stress response, toxin and toxins, amongst others, indicating that these persister cells probably use or they employ coordinated, multifaceted stress response stress adaptation responses, and also that they have distinct gene expression signature compared to the stationary phase cells. So to help us appreciate that persistence do not use resistance mechanisms, we looked at some of the ciprofloxacin-associated resistance genes.
Please,
and
if you can wrap up for us.
Okay, and here we focused on the fluoroquinolone targets, which include gyrase A, gyrase B, and para-A. And these genes are actually not, actually remain unchanged, except for gyrase A, which is actually upregulated in both the persister cells and temperature-tolerant cells, which means that it's probably just one of the stress response mechanism pathway. So we also looked at the toxin and toxin systems and stress response genes, and here there are multiple of the TA systems that are expressed, and here we can see that the temperature tolerance actually have more TA systems that are expressed, which means, or which could indicate that they are probably using TA system as one of their mechanisms, which could contribute to what we observed phenotypically, how heat stress actually confer persistent formability on the cells. So then this is just to show that some of these genes that we identified, we have some that haven't been associated with persistence. So in summary, we've been able to identify that the clinical and environmental E. coli isolates form persisters under the ciprofloxacin and meropenem antibiotics, and that heat stress promotes persistent cell formation, and that the persistent cells show distinct transcriptional profiles relative to the stationary phase cells, and that their persistence is majorly driven by metabolic reprogramming and not resistance gene activation. We also identify that E. coli persisters remodel gene expression by activating toxin and toxin systems and specific stress response pathways for survival. So the clinical significance of this is that we understand how persistence can contribute to recurrence of infections, and persistent cells, even though they are non-resistant, they are stress tolerant, which means that prior exposure, especially in infected patients, can contribute to their survival under antibiotic stress. And current diagnosis usually miss persisters. So understanding this can help us contribute to or lead us to better treatment strategies. And just like what my findings have shown, that we have potential targets that we can use as anti-persistent therapy and future diagnostic in clinical settings. I'd like to acknowledge my supervisors. I'd like to acknowledge my supervisors, WACCBIP, and my lab members. Thank you all for listening.
Thank you, Tosin. 1, 2, 3. Okay, and 4, right? Please make your questions short for us so that we can get time to answer them. Thank you.
Okay, thank you for your presentation. Very good one. My question is about the gene that was still expressed and the persistent ones, as you mentioned, the resistance to antibiotic. So did you consider knocking them off to see if the effect persisted, if they were still tolerant to the engagements that you used?
So do you mean knocking off the genes,
yes,
the fluoroquinolone targets?
Yes.
I mean, that's very interesting. However, I mean, maybe for my postdoc, because I'm actually, yes, my PhD.
Good.
Okay, thank you.
Thank you, Tosin, for this really beautiful presentation. I was really curious to understand why you had to just draw that conclusion, number 4, as regard persistence associated to, though that's your finding, metabolic reprogramming, and then boldly is not connected to genetic resistance. So I wanted to know if there's any possibility of extending this work to having that clear-cut answer why that genetic, influence is likely not really contributing to what you observed.
Okay, so how I'm going to approach your question is that we are trying to understand difference between resistant cells and persistent cells. So we understand that for resistant cells, they use genetic mutations and also acquired resistance genes. And although the isolates that we use actually have some resistance to, or actually possess some resistance genes. However, if you look at the expression that I showed, the target of fluoroquinolones were actually not changed, like they remain unchanged in both the control and the treatment, except for the gyrase A. And the gyrase A is also upregulated in temperature-tolerant cells, indicating that it's possible that this gyrase A is, the mild upregulation might be based on one of the stress mechanisms. Maybe there's a pathway that can trigger the expression of this gene. So the persistent cells are clearly distinct from resistant cells. Although, I mean, there are recent studies now that are actually looking at how persistence can result in evolution of resistance. But that's like when you do like continuous exposure of these strains to antibiotics.
Thank you.
Yeah.
Comments. There are two observations. Please, when do you have a demarcation between persistence and resistance? Because why I'm asking this is, yes, you did say it's phenotypic, but we all know that at some point from the phenotypic, it may go down to genetic. Then the second observation is, in your control, why didn't you maybe choose a control, like a positive control for the resistance gene? You're having the normal susceptible, then you're now having persistent. Why don't you now have like a resistance so that you will know if truly your persistent hasn't or is not giving you the same response in gene expression level with your resistance? Maybe that should be something of consideration, please.
Thank you so much for your question. So I think my explanation can actually answer your two questions. So for persistence, you need to use like concentration of antibiotics that would allow the cells to be in their susceptible form. So when you have resistant cells and you check their minimum inhibitory concentration, this is a concentration that can kill about 90% of the cells. So I determined that, and then I was able to identify their minimum inhibitory concentration, and I used ×2 of that, like double the minimum inhibitory concentration, in order to be sure that the cells are actually not there because they are resistant to the antibiotics. And to also add to that, after exposing them to this ×2 of the antibiotics, we sometimes check their minimum inhibitory concentration to be sure that they still have the same minimum inhibitory concentration as what we observed before introducing them to the antibiotics. So for resistance, when you introduce them to antibiotics, their minimum inhibitory concentration will continue to increase. I don't know if you understand. But for persistent cells, it remains the same. So it's like a transient phenotype. It's something that changes. So if you have your persistent cells and you don't immediately, if you have a subsequent assay that you want to do, you need to do it like immediately before they start regrowing or, like switching back into
we
their
take the last
susceptible
comment or
phenotype.
question. Thank you, Stosen.
Thank you very much.
Okay. Let's take this one.
Very nice talk, and I appreciate the challenge to study persistence. Just to put my question in context, I mean, persistence are really the reason why TB takes 6 months to be treated. Because in the first 2 weeks, you actually kill 95% of the bacteria, but you need 5.5 months to treat persistence. But one thing that I want you to appreciate in your approach, e.g., when you try to translate your data into clinical relevance, is that this subpopulation are existing before you treat this patient. So they will be induced by other means, such as the immunology of the patient. Yeah. So, because, I mean, that's why maybe they will be higher in case of sepsis compared to other infection that you described. But in none of your approach, you account for this immunological component that may be triggering the emergence of this persistence And that's something that I want you to appreciate. Because towards the end of your talk, you were alluding to some genes that could help to detect this persistent, and I asked myself, how are you going to translate that? Because, I mean, there are subpopulation of the bacteria. Are you saying that you come up with a diagnostic that will detect persistent among your population to treat them differently with the different antibiotics? Really, try to reconcile this with regard to the output of your finding into translation.
So in terms of diagnosis, so there's an assay that we do in identifying persistent cells. We call that tolerance disc test. So this is a bit similar to antibiotic disc diffusion test for resistance. And so for this test, it is something that is applicable in clinical settings. It's just that it needs a bit of modification, because when you do the normal disc diffusion test, after 24 hours, if you change the antibiotic disc and replace it with a glucose disc, persistent cells are actually going to grow. So although you have like zone of inhibition without any colony initially, after 24 hours of replacing your antibiotic disc with glucose disc, you're supposed to see, if truly they are persistent cells, you're supposed to see like new colonies that would be growing, indicating that they were actually not growing because of the presence of antibiotics. But immediately you stop the treatment, then they continue to proliferate.
Thank you very much. I think this discussion can continue. There's a question online.
Yes.
Can we take that and then we can wrap up? Thank you.
The question is from Joseph Yaya, one of our PhD students. It says, can persistence lead to resistance and vice versa?
Yes, please. I mentioned earlier that there are some studies that are looking at evolution of persistence into resistance. But can resistance lead to persistence? Yes, I think because continuous exposure to antibiotics can also result in tolerance of the bacteria to antibiotics. It can lead to tolerance, but maybe not persistence.
All right, thank you so much, Tosin. So we have come to the end of the section, and we want to let's appreciate all the 4 speakers. Thank you very much.
Yeah. Okay. So we thank our chairs very much for the excellent moderation. And whilst I called the last 2 chairs to chair the last 3 talks, there was a question that we missed online, and the question was for you, Serge. If I can read it real quick. Before that, so if I may ask Dr. Lucas and Dr. Jesley Chirawa to come and chair the last session. Now the question, how are these septin cages assembled, and does actin polymerization or microtubule assembly play a role in this process?
It's a wonderful question.
Please come in, if you don't mind, on the mic.
So the question is, how do septin cages assemble, and what is the role of the other cytoskeleton components? So we think about this question a lot in my lab. It's a complex procedure. We're studying it in depth. It seems to require at least groups of 4 septins to polymerize into filaments. We can make cages in vitro in the absence of other cytoskeleton components, suggesting that kind of the basic septin cage can happen without microtubules and actin. But inside cells, we have observed that actin plays a key role. I have not yet observed a role for microtubules, but I strongly suspect because septins can interact with the other cytoskeleton components, it will play a role in the ultimate cage, but we can make a basic one in vitro in the absence of all of these other things. Is that okay for now?
Hope so.
Thank you. I'm happy to talk about it, whoever asked.
Okay. Oh, there's a second one as well, but I guess maybe it's asking what the limitation of zebrafish model is, but maybe I don't know.
There's zero limitation.
There's zero limitation. Okay. So thank you so much for that, and thank you for chairing this last session, Dr. Amenga Etigo and Dr. Chirarua. So we have 3 talks, and I will hand over to you to take us home.
Well, thank you for the opportunity given us, myself and Jessie, to chair the last session. And so we have the first talk from our own Dr. Collins Muranga. He's going to give us a talk on collaborative efforts in malaria parasite genomics. I think Peter put the best talks at the last minute.
I should be a politician.
You have your audience. 15 minutes.
Yeah, so thank you very much for this opportunity. My name is Muranga Collins, and I'm going to talk to you about a collaborative effort that we are making to be able to study, try and study mosquito-derived oocyst samples and see the levels of genetic diversity and population structure and how this influences their adaptation in mosquitoes. So just to start off is that a lot of work has been done. There has been established analysis of different parasites from across not just Africa, but other populations in Southeast Asia. And one of the things that they've done so well is that they've been able to sequence up to right now about 20,000 or more parasites. And they've been able to establish that parasites, depending on the geographic location, they tend to form different population structures. The only difference is that most of this analysis has been done in parasites from blood samples. And what we know is that parasites from blood samples, they tend to demonstrate high levels of multiplicity of infections. And this is key to what we are going to study, in that parasites close to 1 here with an FWS close to 1, they are monoclonal parasites. And then parasites around this region are highly polyclonal parasites. So within an infection, you can find the individual having more than 1 clone of the parasite. And this kind of translates to what we want to look at, whereby if a mosquito bites those individuals, depending on the type of parasites that they harbor, so if they have monoclonal parasites, the gametocytes that are going to be picked are going to be highly clonal. And if an individual has probably parasites of the same clone with a few mutational differences between them, and then sometimes you can have parasites that are highly diverse. So probably the mosquito has picked parasites that are totally different. And this presents a challenge in that these parasites, depending on that particular background, they are either going to have self-fertilization. These are parasites with similar clones, or they are going to have some form of inbreeding. These are parasites with slight differences between their genotypes. And sometimes you can have cross-breeding for parasites that are highly diverse. And what happens is that these parasites go on to undergo fertilization. And between formation of the zygote and the next step, which is the ookinete, there's a series of stages, meiotic stages that happen. Although meiosis in parasite happens post-fertilization, whereby formation of the zygote undergoes an initial duplication of the DNA through pre-meiotic replication, which forms kind of a tetraploid cell with 4N DNA. And this goes through several stages of chromosomal segregation to produce an okinase, which has a 4 haploid genome. And this haploid genomes then go on to crossover through the into the basal phase of the mosquito, whereby they form the oocysts. So the oocysts are developed for about 14 days, and they undergo endomitosis or sporogony to produce sporozoites, which eventually invade or are injected back into the humans during the next biting phase. This whole process takes about 21 days, but it's not very clear what happens in the oocyst development stage. But we can utilize this to be able to study, because we have high copy numbers of DNA. We can use this to be able to infer the diploid or the heterozygosity levels of this initial parasites that were picked. And this genotype and heterozygosity patterns, we can use them to derive the type of haploid or diploid genome that was formed, and eventually be able to study the genetic diversity and population structure of parasites in different endemic regions. And hopefully this can help us, start to study initially things like adaptation of different parasites to mosquito vector species. So what we've done so far is we've developed through a pilot project, but we've developed a sequencing strategy to generate genomes from Plasmodium oocysts, and by bioinformatics strategy to be able to analyze these genomes. And we've already started looking at patterns of sequencing coverage and heterozygosity across the genome, so that we can be able to investigate the genomic variation. So basically, once mosquito oocysts have been collected, we extract DNA, and this is processed through selective whole genome amplification, and we end up having libraries that we sequence here at WACCBIP. And we've also developed a robust bioinformatic process where we do quality control mapping of these genomes, and eventually variant calling to be able to generate variants that we can analyze. This work, as I mentioned, is a collaborative effort. For pilot analysis, we are using samples that were collected earlier on through Thomas Anthony, and they collected these samples from Tanzania, and they've helped us to sequence them and develop some of our sequencing and analysis pipelines. Currently, what we've been doing since 2024 is to sample across Ghana with a collaboration with Professor Afrane, where they sample both indoor and outdoor in these 2 locations, southern Ghana and northern part of Ghana. And they've collected about 6,700 Anopheles mosquitoes. They dissected about 4,403. And we were able to get 39 positive mosquitoes with about 154 oocysts. So we are currently processing these samples to be able to study the genetic diversity and population structure of these parasites. But for the pilot samples that we sequenced, what we were looking out for essentially is to be able to capture heterozygosity of the parasites. And and what I'm showing here is just a representation of how reads are mapping to the genome. And we can see that we have sites where we have very clear heterozygous mutations that we can be able to now tell that it's possible that some of these oocysts became from parasites that kind of have diverse background, and they've cross-fertilization between parasites that have a diverse background. And you can see this in different chromosomes or even within the same chromosome, whereby you have very clear heterozygous with almost 50% kind of distribution of reads covering those heterozygous sites. So so that's just looking at individual sites, but you can extend this to be able to look at across the whole genome, whereby you can see that the yellow represents homozygous SNPs and the blue heterozygous SNPs. And you can see that across each individual chromosome, some oocysts have relatively fewer number of heterozygous SNPs, while others have quite a number of heterozygous SNPs across the entire genome. Although there was differences in terms of coverage between the different oocysts and the total number of SNPs that we were able to identify. So what we thought about is that you could have some SNPs that are likely to have like an allele frequency the number of reads being, or the proportion of reads that are supporting the alternate mutation being very low, and this could represent that it's just a homozygous reference. So and then you could have a high number of reads that are supporting the alternate and very few reads that are supporting the reference allele, and this could probably be a homozygous alternate read. So we decided to look out for the allele balance of these mutations so that we can filter only mutations that, for the heterozygous mutation that are likely to be true in terms of heterozygosity or being from different backgrounds. So we did this. We filtered out all the SNPs in the blue, and we kept only SNPs within that have a good proportion of allele balance greater than 0.2 up to 0.8. And then out of the SNPs that have been identified within malaria genome, and this is really to try and see whether we have SNPs that are likely to be artifactual. So you can look at a database of about 20,000 SNPs, because we are thinking if a SNP has been seen before, is a truer mutation compared to mutation that we are identifying now as new mutations. So we integrated our SNPs with malaria gene data sets to be able to filter out those SNPs that are likely to be malaria gene kind of validated SNPs. Here I'm just showing a table of all the different oocysts, the total number of mutations that passed our QC criteria, and you can see heterozygous SNPs. So this is, we were hoping to see this kind of distribution where you have some oocysts that have high numbers of heterozygous SNPs, while others have a low number of heterozygous SNPs, and some have like intermediate heterozygous SNPs, because of what I had already described earlier, that from polyclonal infections and monoclonal infections, you expect to see this kind of distribution. And when you filter out all these heterozygous SNPs with what has already been presented before within malaria gene, then you get a more truer picture of the heterozygous mutations that we are getting within our oocysts. And here, what you can see is that we have a set of mutations, a set of oocysts here that are highly, almost all the mutations are homozygous, which represents that probably the clones were highly monoclonal parasites. And you have a distribution of oocysts here that have a good number of heterozygous SNPs, and others that have fewer heterozygous SNPs. And this could just represent some form of inbreeding within parasites that have very minor differences between between them, while here is more of self-fertilization between parasites of the same clone. And then here you have like parasites that are presenting with cross-fertilization between parasites with diverse background. So, so really this gives us like a proof of concept that we can be able to derive parasites that have like a diploid genome background from oocyst samples, and we can be able to delineate all the expected populations depending on, especially in wild-caught mosquitoes. So with this, just to summarize, is that we've seen some level of heterozygosity, good distribution, although the heterozygosity levels is low. So we are hoping that if we sequence more oocysts, we'll be able to see a better distribution. And using malaria gen, we've shown that we can have validated proportions of heterozygous SNPs, which is really the first kind of unbiased heterozygosity estimates of oocyst samples. But there's need to further test and kind of modify the SWGA protocol so that we can see whether we can be able to improve the genome-wide coverage and be able to get parasites with higher proportions of heterozygous estimates. Currently, we've sequenced a few more of the pilot samples from Tanzania, which we are currently analyzing, but we hope to sequence the samples from Ghana, which are currently being processed, and so that we can be able to compare the population structure and genetic diversity of parasites from different mosquitoes and different endemic regions. And we'll be able to see things like does the adaptation of some of these parasites to different mosquito population or Anopheles, whether it's Anopheles gambiae or Anopheles fenestus. So thank you very much. I would like to acknowledge my advisors, Professor Gordon Awandare, Dr. Lucas, and Prof. Conway, Prof. Afrani for the collaboration, and Joyce for facilitating the sequencing, and this whole team of people who have contributed to this work, and Wellcome Trust for the fellowship. Thank you very much.
Two burning questions.
Okay.
Thank you for your presentation. Mine is just a comment and observation. Please, regarding mosquito samples you have, you said you did dissect, and I believe you got the oocyte from them, right? The sample you had, is it per mosquito sample, or you pull them together?
The
oocyte you got, yeah, oocytes.
Per mosquito sample.
How are you able to get separate the oocyte from one mosquito? Because I'm doing kind of a similar work. It's kind of before you could get them, sometimes they dry up on the slides. I don't know, maybe you are using a more sophisticated equipment than I am doing at the moment. So please, just a curiosity.
Are you trying to isolate oocysts?
I have done that. I knew the stress we went through. So I don't know, maybe you have a more automated equipment, because I kind of did it in a crude way. I kind of found a way, I optimized a way in which we could get the oocytes from the dissected mosquito. But I was just curious, because I know having it from one mosquito is kind of a trouble. Then I don't know. Thank you.
Yes, I think we can meet and discuss more about the isolation of the mosquitoes. Yeah.
Yes. Any other question?
Okay. In your identification of SNPs, were you able to assemble hypervariable families?
We haven't done the hypervariable gene families, but I think it's something we'll do once we've optimized all our sequencing protocols. Yeah. But the assemblies are there. It's just to look at the hypervariable regions. Yeah.
Thank you, Collins.
Thank you very much.
Wonderful presentation.
Okay, so the next one is a fellow session, Etel Deborah, who will be speaking on isolation and characterization of bacteria-eating viruses in Ghana. Oh, she's speaking online. Okay, she's in Mass. Yes.
Hello, can you hear me? Yes. I hope you can.
Hello. Yeah, can you speak up?
Hello, can you hear me, please?
Yes, that's better.
Okay, so good evening. My name is Ethel Deborah, and I'm presenting my progress on the project Isolation and Characterization of Bacteria-Eating Viruses in Ghana. So AMR has become a global pandemic.
Sorry.
And
Hello. Can you please put on presentation mode?
Okay. Yeah. I think it is.
No, it's not on presentation mode. Right.
Is it now?
No. Can you please exit and start again?
Okay. I just did. Stop share. Hello? Can you see it now?
It's still not in presentation mode.
Oh. Maybe. It's showing as presentation mode here, so
Yeah, select the slideshow. Yeah.
Is it working?
Okay.
Because I just did.
Okay. Maybe you should just proceed.
Okay.
But it's still not in presentation mode.
Because I have selected the slideshow. Anyway, sorry about that. Good evening once again. My name
share your screen.
Is the screen showing?
Yes, we can see. Okay.
Hello.
All right, please continue. Yes. Sorry.
So I'm presenting my progress on the project Isolation and Characterization of Bacteria-Eating Viruses in Ghana. So AMR has become a global pandemic, and review reports have forecasted that bacterial AMR would cause 39.1 million deaths between 2025 and 2050. So efforts are being made to develop alternative therapeutics, and one of these is phage therapy, which is a therapeutic administration of bacteriophages to eliminate bacterial pathogens. And phages are viruses that specifically infect bacterial cells. So a phage infection typically involves the A phage infection typically involves the recognition and binding of the
Hello?
phage proteins. Hello?
Sorry, we can't see. The slides are not moving. Yes.
Then it's probably the internet because mine is moving.
Hello, Ethel. Hello, Ethel.
Hello.
Hello. Do you have two screens that you're using?
No, it's just one.
Hello, Ethel.
Well, the slides we see here.
So please, can you stop sharing? Then when you're sharing, you select screen. Okay, so go to share. So when you are selecting the options,
Okay.
Yes. Yeah. Yay.
Sorry about that. I have to start again, I guess. Okay. So I'm presenting my progress on the project Isolation and Characterization of Bacteria-Eating Viruses in Ghana. So AMR has become a global pandemic, and review reports have forecasted that bacterial AMR would cause 39.1 million deaths between 2025 and 2050. So efforts are being made to develop alternative therapeutics, and one of these is phage therapy, which is the therapeutic administration of bacteriophages to eliminate bacterial pathogens. And phages are viruses that specifically infect bacterial cells. So a phage infection involves the recognition and binding of phage proteins to bacterial cell surface receptors and the injection of the phage genome into the bacterial cell. From this point on, the phage begins its life cycle, and phages can be classified according to their life cycle that they would undergo. The first is the lytic cycle, where the phage immediately produces new viral particles and then lyses the bacterial cell. The second is the lysogenic cycle, where phages integrate their genome into the bacterial cell and just stay dormant until lysis becomes critical. And the third is the chronic cycle, where phages produce new viral particles, but here there's no cell lysis, so the new particles just leak or they are extruded out of the cell. Now, not all phages that are isolated can be used therapeutically. So this means that you should look out for certain traits to enable the selection of the right phage for therapy. And some of these traits would include, first, the lifestyle. We want phages that are strictly lytic because we know for sure that these ones are going to do the job. They won't slack. And you'd also want to look at the host range, depending on the kind of infections you'd want to use the phage to treat, because phages are very specific for their bacterial host. And you want to look at the stability of these phages under varying temperatures, pH, and even other solvents, because they are composed of proteins that can easily be denatured, which would inactivate the phage. And you'd want to look at how fast the phage multiplies, because the faster the phage multiplies, the more effective it is when clearing its bacterial host. And you want to exclude the presence of any toxin encoding genes or AMR genes within the genome of the phage that can be transferred to bacteria. Lastly, if your phage has antibiophone potential, it would be an added advantage. So this would form the basis of phage characterization, right? Now, there are different ways that phages can be used in therapy. The first is the conventional phage therapy, where a single phage formulation or a cocktail of phages can be administered. Or in the second one, the phage lytic proteins could be isolated and used apart from the whole phage particle. And there's also the scenario where phages are combined with antibiotics, which is usually seen in clinical practice. And this has been shown to have synergistic effects. And lastly, phages could be engineered to carry therapeutic genes or antibacterial molecules. So this brings me to the rationale of my study, which states that there's been reports of the persistent emergence of multi-drug resistant critical priority pathogens in Ghana. And this means that we need to look out for alternative therapeutic approaches for these resistant pathogens. And looking at the history of phages, we proposed phages as a promising strategy for the control of these resistant pathogens. So my study was guided by 3 questions. The first is, will environmental phages show antibacterial activity against MDR bacteria? 2, what conditions are favorable for phage survival, stability, and bacterial infection? And 3, will phage antibiotic combinations increase antibacterial activity against MDR bacteria? So the overall aim of my study was to isolate and characterize phages specific for 3 escape pathogens. And specifically, I sought to isolate these phages, characterize them, and examine the efficacy of different phage antibiotic combinations against MDR bacteria. So of course, to start with the phage isolation, I collected 74 environmental samples, and environmental samples because phages are found everywhere in nature where bacteria exist. And I used 3 MDR, archived MDR strains in addition to 1 E. coli ATCC as host strains for phage isolation. Then I went ahead to use the plaque assay to isolate these phages. So at the end of the isolation stage, got 30 phage isolates with majority being isolated for or being specific for Klebsiella pneumoniae. Now looking at their plaque sizes, the plaque sizes varied between 3 millimeters in diameter and less than 1 millimeter in diameter. So from this point on, I had to select 2 phages to go ahead and characterize further. So I selected the Klebsiella phage that had the highest or the largest plaque size, and then the E. coli ATCC. So from this point on, the Klebsiella phage will be referred to as KPP1 and the E. coli phage as ECP2. Now looking into their plaque morphologies, both phages, that is KPP1 and ECP2, produce a kind of plaque referred to as the halo plaque, and this is seen as a clear zone in the middle which is surrounded by a turbid region. This turbid region is associated with the presence or production of depolymerases that are known to break down or degrade biofilms and capsules, which the phage does to enable access to bacterial cell surface receptors for further access. Yeah. So looking at the phage morphologies, the morphology of the phage itself, both phages, that is KPP1 and ECP2, had a cyphovirus morphology, which is seen as an icosahedral head with long flexible tails attached to tail fibers for binding. Now, it's possible to observe the lytic activity of phages in vitro, and this is done by infecting an actively growing bacterial cell culture with phages and shaking for a couple of hours. Then you'd now see that there's a decrease in the turbidity of the bacterial culture, and this can also be seen on when you do it on the plate as well using the plaque assay. So I wanted to see how fast the phages multiply with time, and I did this by infecting the bacterial host with their specific phages at different MOIs, which is the ratio of the number of phages to the number of bacterial cells. And generally for the 2 phages, it was observed that as the MOI increased, the time it took for the phages to clear or reduce the cleared bacterial cells decreased. And it was interesting to know that even for the lowest MOI of 0.01, there was a clear difference in the growth of the phage infected and phage uninfected bacterial host. The next thing I did was to look at how fast these phages actually multiply within the host. This was a function of the latence period, which is the period between the infection of the bacterial cell and then the release of the viral particle. So within this time, you expect there to be DNA replication, transcription, translation, and assembly of the whole phage particles. And this was between 15 minutes and 25 minutes, which is short or faster compared to other phages that would infect similar bacterial hosts. And now looking at the stability of these phages under varying temperatures, the phages were incubated under these temperatures for about an hour, and it was found that they were most stable between 4 degrees Celsius and then 37 degrees Celsius, although when they were incubated at -20, 60, and then 70 degrees Celsius, there was a rapid loss of the phage concentration. And looking at the stability of these phages under pH between pH 1 and 11, it was interesting to note that although the phages were inactivated at pH 1, right, when they were incubated between pH 3 and 11, there was no loss in the concentration of the phages compared to the phage buffer, which was used as a control. So, and this is important because most phages have been found to be more stable between pH 5 and 9, but then anything beyond this, they would be inactivated. Similarly, the phages were incubated in sodium chloride solutions up to 30%, and it was also found that they were quite stable up to 25%, which, looking at this trend of pH and sodium chloride, their stabilities in extreme pH and then sodium chloride concentrations, it could infer that these phages probably have certain structural adaptations that protects them to a certain degree from extreme conditions. So the next thing was to look at the host range, that is how many other bacterial pathogens within the escape group, apart from the original isolation host, could these phages infect. And although the phages were not able to infect the other members of the escape pathogens with at this point, right, they were still quite broad, because if you pick Klebsiella phage, for instance, it was able to infect another strain, another species, and even E. coli, which is telling us that they have quite a broad host range. And sequencing the genome of the phage, the classification identified these phages as strictly virulent, which is what we want. And they were also classified under the genus Weber virus. And these phages that belong to this group are known to be specific for Klebsiella bacteria under the genus Klebsiella, which explains the host range that we saw in the slide before. Now, the genes within the genome of the phage encoded proteins involved in DNA synthesis, transcription, and repair, in the packaging of the an assembly of the whole phage particle. The genes also coded for proteins involved in host cell lysis, and then there was this counter defense mechanism that can be used against bacterial restriction modification systems. So it's important also to note that no resistance or virulence factors were found within the genome of both phages. So for the final part of the work that I did.
Tell. Yeah, sorry. Please, you have just 2 minutes left.
Okay, it's fine. For the final part of the work that I did, phages were we had this phage antibiotic combination where the phage, different MDR bacteria were infected. Yeah, so we treated different MDR bacteria with a phage antibiotic combination at these concentrations with these 4 antibiotics. And we found that even for both phage susceptible and phage resistant strains, when these combinations were used, there was a significant increase in the level of susceptibility of all the MDR strains that were tested. And a couple of mechanisms have been suggested to explain this activity, but I guess would have to investigate it for each of these strains because they could vary. So in summary, all that I've said so far, KPP1 and ECP2 are strictly lytic, and they multiply and release new phage progeny relatively faster. It was found that they had a broad host range, and they are most stable between 4 degrees Celsius and 37 degrees Celsius. They are also stable between pH 3 and 11 and can withstand salt concentrations up to 25%. We are also suggesting that looking at their plaque morphologies, these phages may possess antibiofilm potential, and they did not possess any virulence or AMR genes. And we are also saying that these phages could boost antibiotic efficacy even against non-host MDR bacteria. So this implies that KPP1 and ECP2 could be considered as potential therapeutic agents for treating infections caused by MDR, Klebsiella pneumoniae, and E. coli. These agents could also boost antibiotic efficacy towards non-host MDR bacteria. And finally, the lytic proteins that were found in the genome could be isolated and used separately or in combination with antibiotics as antibacterial agents. So I'd like to acknowledge my supervisors, our collaborator, the AMR Research Group, and my funders. Thank you for listening.
Thank you very much. Guess we'll have time for one question. One question. All right. Okay, Etel, thank you. It looks like there are no questions. Thank
Okay,
you.
thank you.
Well, so we switch gears back to malaria. We have Nana Mwako, Minister, to so he's going to be, his topic is truncated in the program, but I think he's going to talk about Anopheles clusiae and insecticide resistance and adaptation to bioclimatic across different bioclimatic zones in West Africa.
Yes. Okay. Good day. I'm Kofi Amwako,
You have
Enoch,
15 minutes. 15
well known as Minister. So I'm here to present on genomic population structure and insecticide resistance mechanism in the malaria vector Anopheles clusiae across contrasting bioclimatic zones in West Africa. So the malaria vector Anopheles clusiae is an important contributor to malaria transmission in West Africa. Among the Anopheles gambi complex, this vector is believed to be better adapted to specific bioclimatic environment across variable ecological landscape in West Africa. West Africa's diverse environment affects the behavior of these mosquitoes. Insecticide resistance makes control tools like these bird nets and IRS less effective. On this note, the problem here is that there is restricted gene flow and varying selection pressures that may drive resistance distribution. And it is important to study the genetic dynamics, gene flow, and to track the resistance across West Africa using whole genome SNP data and inversion karyotypes. In this study, we focused on 1,324 whole genome sequence data of Anopheles clusiae from 2016 to 2018 from the northern Ghana. And also, we looked at 475 whole genome sequence data from the southern Ghana, including samples from Burkina Faso, Mali, and Cote d'Ivoire. Mosquitoes were sampled between 2016 and 2018 using human landing catches, CD traps, and spray collections. We performed whole genome sequence analysis and provided SNPs data and inversion karyotype. So in this work, you're expected to see PCA, which is the population structure, neighbor joining tree, FST, and look at resistance, both target and metabolic, and also some selection scans. To investigate the population connectivity across Anopheles clusiae in West Africa, we focus on PCA, and we look at regions in Ghana and other countries. So we can see from the regional level that there are two main clusters, the northern cluster and then the southern. And the neighbor joining here depicts how gene flows between the southern clusters. And we can see that there is restricted gene flow taking into consideration the northern and southern comparison. Let's look at across countries. We can see that in the northern Ghana, Ghana shares borders with Burkina Faso and Mali, and we can see more diversity here, as well as the southern forest, where we are considering the South Cote d'Ivoire and Ghana. To further confirm the similarities, we perform PFST, where we can see that there is high conductivity among southern population, and there is restricted gene flow within northern and southern. Now, there is high FST between the northern and then the southern. And then within the northern cohort, we can see that Burkina Faso, Mali, and Ghana have lower FST. It means they share similar genes, and you can see from the neighbor joining tree here that you can see there is gene flow happening here. To further investigate the gene flow of Anopheles colusia in the Central and West Africa, we perform principal component analysis on 2 inverted chromosomes. And here we are focusing on the 2LA and then the 2RB. Now, within the regional level, we can see that we have 3 main inversion karyotypes that is happening here, and this is mainly focused on the southern forest. And we can see that there is gene flow happening within here. We have the inverted, where it mostly happens in the dry areas, and then we have the standard, where it mostly happens in the wet environment. And then we have the heterozygotes, that is the mix between the inverted and standard. And we can clearly see that there is restricted gene flow within the northern and southern on chromosome 2RA. Now, when we come to the across countries, we can see that within the northern savanna, where we shared border with Burkina Faso and Mali, we can see that there is unrestricted gene flow in the dry, arid regions of Ghana, Burkina Faso, and Mali. That means there is gene flow happening here. Looking at the 2RB, we can see a clear cut that now these 3 inversion karyotypes is happening in the northern savanna. And then we can see that within the southern, we can see that there is restricted gene flow between the northern and southern, but there is unrestricted gene flow happening in the southern forest, which is mostly the wet and forested. Now, looking at across country, we can also see that we can still see the 2 main clusters and the inversion karyotypes. Now, the southern forest, which is mostly Côte d'Ivoire and Ghana, we can see that there is unrestricted gene flow. That means mosquitoes are able to move in and out, and they carry this. To look at the insecticide resistance, we are looking at the target site and the metabolic. With the target site, we are focusing on a minor allele frequency of just 3 genes. And here we can look at the VGSC, we are looking at the LDL, and then the ACE1 gene. Here we can see the KDR allele mutation, which is the L995 mutation, which is fixed across all population, and which is linked to resistance to parathyroid and DDT. And we can also see the double mutant, that is the V402L and I15L2, which is also increasing. And this substitution pair is expected to replace the L995 mutation over time. Now, we also observe the allele pair, that is the A296G and T35M, as well as the A29S and T345A at the DRDL locus. Now, this mutation confer resistance to organochlorines like deldrin, and we can see that the A296S and the T45S is mostly focused in the northern part, and the A29G and then the T345M is mostly focused in the southern part. Studies have shown that, up to date, this specific mutation can only be found in Burkina Faso, and since Ghana and Burkina Faso share the same borders, these mosquitoes don't need passport to cross over. So, looking at the metabolic, we focused on copy number variations that are associated with insecticide resistance. So we are looking at the cytochrome P450, we are looking at carboxyl esterase, and the GST. Okay, so here we can see amplification of the CYP1 and CYP2 gene, which we can see here, and we can see that these are more high and they are more resistant to pyrimidine methyl, and this has the ability to detoxify insecticides and parathyroid. Now, looking at carboxyl esterase, CNVs, we can see that it is also increasing, and it is also linked to pyrimidine methyl resistance, and these act as a detox system to neutralize poison. We can also see the GST. So we can see that these genes are more dominant within both northern and southern, and these genes, these mutations actually help these mosquitoes to survive exposure to these chemicals that we use. So looking at signals of selection, we focused on chromosome 2RL, and we can see both from the northern savanna and southern that we have this GST metabolic gene, which is found within both population, and this has the ability to neutralize insecticide resistance, like detamethrin in bed net and permifos in sprays. And here we have heavy insecticide resistance used in both dry and wet savannas, which are mostly driven by a rapid spread of these insecticide mosquitoes. I know there are interventions that have been happening in the northern Ghana, but we can see within the southern we have KIP1, which is only found within the southern, and most people within the southern forest uses these aerosols, and they use most of this insecticide spray as compared to the northern sector where they have this intervention, which is mostly more of agriculture stuff. Yeah. Okay, so we also focused on chromosome X, and we can see that this CYP9K gene also shows strong positive natural selection within both population. And this CYP9K gene has the ability to, these mosquitoes have these genes, so it is hardly for them to die. So there is the need to focus on new ways where we'll be able to combat this resistance, this metabolic resistance. So with observation, we're able to see a restricted gene flow between the northern-southern comparison in Anopheles closii population, but we saw an unrestricted gene flow across countries. In the north, we can see higher target site parathyroid and metabolic cross resistance due to IRS by NMEP. And then within the south, we can see a unique resistance mechanism linked to varied insecticides used by household. And the environmental condition actually limit the gene flow, shaping the distribution of insecticide resistance alleles. So on that note, it's important to tailor malaria control strategies needed to address these at the regional resistance patterns. So I would like to acknowledge the founders and also my PI, Dr. Lucas, Kelly, Prof. Wandare, Chris, and Alistair, and all my WACCBIP teams. Thank you so much.
All right. Questions?
Thank you, Minister, for malaria, right?
Yeah.
Okay. Yeah, okay. I really enjoy your talk, and it was quite interesting. But mine is just maybe commendation or maybe comment. Have you bothered maybe to corroborate your data with maybe going to actually check if these markers are actually responsible for these resistance? Please?
Yes, so with these markers, so literature has actually proven that these markers are actually there and they are working. So with our selection scan, we were able to see similar peaks within.
Yeah, actually, they were. I was just thinking maybe if you can just, it will be lesser work compared to what you've done. Maybe to the southern region, if you go to the southern forest, the marker that you see to be specific to that region. Maybe just design a little susceptibility test to further confirm the metabolic or.
Yes, I mean that is something
marker.
that is something that we are planning to do. Yeah, thank you.
Thank you for your. Thank you.
One burning question.
Yeah. Well, I was wondering, since mosquitoes in Burkina Faso don't need passports, the ones in Togo, I wouldn't know, maybe they require passports because no, from that part you didn't talk about Togo at all, but you spoke about Côte d'Ivoire, and I know that Togo is also sharing border with Ghana. So I wouldn't know, maybe it was deliberately omitted or it was an oversight. And again, since you are talking about northern parts only, I mean, why then did you bring about, Ivory Coast?
So Ivory Coast shared border with Ghana. That is the southern part.
Yes.
Yeah, so
what about Côte d'Ivoire? Togo?
So we didn't focus on Togo because there wasn't enough data for Togo. Yeah, okay. So we mostly focused on Côte d'Ivoire. Yeah.
Thank you, Minister. Thank you.
Question. Thank you, Minister, for a very brain-stimulating presentation. We are very proud of you. Yes, the question is, you kept on mentioning inversion karyotypes. Did you flip these chromosomes yourself, and what advantage does it give your analysis? Thank you.
Okay, so with the inversion karyotypes, actually what happened is when there is a change in the DNA where a section of the chromosome is being flipped in an inverse form, that is where these karyotypes happen. And research has identified that these karyotypes happens in the bioclimatic areas, and Ghana having such bioclimatic conditions. And actually these karyotypes happens in 3 types. So we have the inverted, that is mostly found within the dry areas, and then we have the standard, which is mostly in the wet areas. And as I said, we have the heterozygote, which is a mix of both. So it's actually it occurs naturally. I don't flip them.
Okay. There's a question online.
Okay.
From Christopher Okeni. What are the possible drivers of the differential gene flows observed in the 2 regions? What are the possible drivers of the differential gene flows observed in the 2 regions?
Good. So the possible drivers of this gene flow between these 2 regions is, these insecticides, look, let's take the northern part for instance. They do most of agricultural work, and there are ongoing procedures that are being in place in the northern part. So they are more of using this DDT within their agricultural activities. But when it comes to the southern part, this is the urban sector where there is a lot of pollution happening, and various household people are using these sprays, right? And there are also car fumes that are happening. So you can see that these things are causing this resistance between causing this resistance. And you can see within the southern sector, even though there was a mix of diversity, mix of gene flow, but we could see from the PCA that at the regional level, Greater Accra was kind of diverging itself. And we know that maybe with time, when we have enough data, we will see a different cluster of Greater Accra as compared as seen in the regional, the southern forest. Yeah.
Thank you. It's been a long day. Thank you, Minister. Thank you.
Fantastic, fantastic. Well, I'd like you to please join me to appreciate our chairs for the last session. Thank you so much. It's a tough session to chair because it's at the end of the day and everyone is quite exhausted. We've had fantastic talks today, and I'm very glad that we are actually not too far away from the original closing time. I appreciate your support, I appreciate your attention, and I hope you bring the same A game tomorrow to listen to another fantastic round of talks. Thank you so much for coming today. Don't forget to tweet key things that you heard that really make WACCBIP stand out strong in the sub-region for the science that we do. And yeah, we will be going to Loggia. If you know, you know. I'm not saying more. So Loggia it is from here, and thank you. See you at 9 o'clock sharp tomorrow. And yeah, thank you to all the presenters. The bus is in front for Loggia. If you know, you know.